Well, good afternoon, everyone, and a very warm welcome to the UKRI Physics of Life, Physics of Brains workshop being held today, the 18th, and tomorrow, the 19th of November. It's the first of our full-scale Zoom workshops of the third incarnation of the Physics of Life network. As well as UKRI and the Research Councils, principally EPSRC, BBSRC and MRC. We're also sponsored by the Rose Trees Trust this time around. And this is also, as well as being a Physics of Life workshop, it's also a Physics of Life and Medicine Rose Trees Trust workshop. We also acknowledge very gratefully the support of the York University-based Life Science Institutes, the Biophysical Sciences Institute and IBRI, the York Biomedical Research Institute, that have helped this along as well. This comes to you through the University of York, and we're also particularly grateful, particularly to Caris Baker, the Physics of Life Administrator, and Steve Parker from Audiovisual, who's helping things run smoothly. I can also announce with great pride and joy, for those of you who haven't heard this already, having mentioned the Institute of Physics, that the Physics of Life community have recently this year been awarded, it was announced last month, the Rosalind Franklin Medal that you'll see there. Not an individual, the Physics of Life network community was awarded it for services in doing exactly what we're doing today, which is to bring people together who have not met each other before, from the physical sciences and from the life sciences, to get around questions and problems that are right on the frontier or haven't even been formulated before. So this isn't a normal scientific conference. If that's a shock to any of our speakers, you have a few minutes to change your talk. It's far more about what we don't know than what we do know. It's about open-ended issues we might take forward. And of course, we'll hear about what people have accomplished in the recent past, because research results are those that are what gives us the questions for the future. Before we get any further, let me just take you through a little bit of information about the work of the Physics of Life network. Really under the, it's important you will know this, because this isn't our grant, this is your grant. It's a very unusual grant. We have about 450K to spend over 3 years from our sponsors. Here they all are. But it's for you to spend, not me or anyone else you see at the front here to spend. It's your grant, and we can fund all sorts of activities. The steering group are very good at turning around applications to those activities very fast. So what do we do as at Physics of Life? We don't have a membership, we have a certain email list, but if you work as a scientist in physics, physical sciences, life sciences or medicine, you are already a member of this community. And you may apply at any moment for funds to support workshops like this one that are open-ended and cover large areas. Sandpits, there's a rather more focused, if you've got a team of people that you might want to build on and do some preparatory work applying for a larger grant application, then we can do that too. And I should say that this Physics of Life network is linked also to in the UK at least, to the UKRI Physics of Life Strategic Research Fund, which next year will open its second call for £15 million of serious interdisciplinary projects in this area. So get your thinking caps on from today. We also run summer schools for early career researchers, where we're essentially building tomorrow's communicating themes and teams. And those summer schools are open to both physical sciences scientists and life scientists at postgraduate and postdoctoral stage. We run under each of our 3-year roles at least one big town meeting, where we invite funders and members of the research community to work together to plan how we support this area in the UK in the future. There are also student bursaries and research pump priming ideas, and everything is free to attend and to apply for. This new tranche, the third time we've rolled, is also, as I mentioned before, under the Rose Trees Trust and under the involvement of the MRC as well, reaching out into the physics of medicine. And this is really our first workshop that includes that angle as well as the life science angle. We've already have some workshops in the pipeline. There are some titles of some of those should be coming up in the next year or so. A couple have been themes we've touched on in the past. Antimicrobial resistance and physics is one of those, very, very fruitful a couple of years back. And as you'll hear from I know Steve Smyth later on, we have a workshop coming up in advanced planning on neurodegenerative diseases, which essentially stems directly from this one. If you would like to find out more about the physics of life, then please do get in touch with Caris. That's her email, Dr. Caris Baker. Also, just about all the information, including forms to use to apply for grant funding for any of those activities. Information on all workshops, summer schools, everything I've mentioned is on the Physics of Life website. It's very easy to navigate. And it also acts as a repository of information on past workshops. You'll find that under Polnet 1 and Polnet 2 tabs. Or you can follow us on Twitter at Physics of Life UK. There's lots of fun going on there. Having said there aren't members, there isn't a member, there is a member of an email list, and that's all you can do. You're already a member of Physics of Life just by being here and doing, even if you're not here. But if you're working in the area, you are still a member. But if you'd like to receive additional information through email, they look a bit like this, then you can register at the website for that as well. These are the co-investigators of the grant. Myself, I'm also joined by Steve Smyth from the University of Leeds, who particularly looks after the medical side of things, and Martin Can, a molecular biologist, cell biologist at the University of Durham. And there's Caris, Dr. Caris Baker, who administers the network. Mentioned, we have one other workshop in the immediate future, early next year, and that's the Rose Trees Interdisciplinary Workshop on Neurodegenerative Diseases. More of that possibly from Steve Smyth, I'm sure, later on, and also on the website. Now, this is the run of today. It's a packed day. I'm really looking forward to it. Please do feel free to use during the talks the question and answer. One of the great advantages about of online meetings is not only that we can fly Bill Bielek in from Princeton and Alec Ballard Robrook later on from Maastricht, and everyone in and out in no time at all, and hear from the very best. But actually, you can also do what you've also always wanted to do and file questions away while you think of them. Pop them in the Q&A facility at the bottom of the screen. We'll have most of the discussions, because the talk slots, apart from Bill's, are relatively compact, 20 minutes. As well as obviously pleading for the speakers to stick to time. Use the 20, but don't get any questions, or speak for 15, and perhaps we'll have 4 or 5 minutes of questions for each particular talk. But there'll be time, in any case, on 4 occasions during the workshop to address your questions and your suggestions to all of the panel who will join me, as it were, on stage during those panel discussion sessions. And I should say, we will plan, although it looks as if this is a 4-hour marathon without even a tea or a loo break, we will stop that panel discussion shortly after 3:20 to give us 10 minutes to make a cup of tea and whatever before Anthony Morland, my colleague, chairs the second half of the afternoon, and we kick off with Amanda Ellison from Durham. So just to remind you about the Zoom etiquette. If you have a question, write in the Q&A tab. That's what it looks like at the bottom of the window, and you can see everyone else's questions. In fact, if you really like someone else's questions, you can also vote for them, and we can get questions moved up the list. finally, and it will be finally at the end of both days, we have a real treat for you. I know one of the aspects of real conferences people are sadly missing is the opportunity afterwards. It all falls rather flat, doesn't it? Suddenly you're not in the conference anymore. You're back in your living room or your office or your bedroom, and you haven't got anyone to talk to. Not with Physics of Life. So during the session, during the afternoon, Caris will give us the link to some software called Wonder, which is Zoom is the equivalent of an online conference. Wonder is the equivalent of the drinks and snacks reception immediately afterwards. You will find yourself as a little icon which you can move on a map. If you know the Harry Potter story, it's effectively the Marauders map. For those of you who don't, it's an icon you can move around a room with tables. You can see everyone else's icon. And when you move yours towards a group of people you'd like to talk to, the software will recognize when you're within range, draw a circle around you, and then pop up a little Zoom breakout room effectively around you. You can talk to them and get to know them and share a chat about the day, and then move off at will afterwards. No downloads required, but if you use Safari, Firefox, or Chrome, it works a lot better. There are the instructions that allow us just to socialize today. It's a bit of a practice. Tomorrow, we're going to use that session for a little bit of work and some play. The first part of the session, 20-25 minutes or so, we're going to be offering you question tables around which you can discuss. People will be there taking some notes, so that we can report back to UKRI on some of the questions and recommendations for grant funding that have come out of the workshop. Then we can have our drinks and nibbles. Unfortunately, you do have to supply the drinks and nibbles, but see you there. I'm really looking forward to those. So I'm going to stop sharing and with no further ado, introduce with great delight Bill Bialek. Bill Bialek from University of Princeton. I've had the joy to know Bill for a long time. Like me, he's a theoretical physicist. His background, but he's been working with experimental neuroscientists, thinking about what they do for a long, long time. But another good reason to invite Bill to talk for us is that he's been very involved in at a high level in the equivalent Physics of Life initiative, the other side of the pond in the USA, and we've had a lot of discussion about how best to bring these sorts of communities together. So Bill, many, many thanks. Take us away on statistical physics for networks of real neurons.
Good. Now I'm both visible and audible, I hope. Thanks, Tom. Thank you, everyone. It is a pleasure to be here. Sorry, just getting the various windows organized. Good. What I wanted to do, the idea that things that happen in the brain are emergent phenomena that arise out of the interactions of large numbers of neurons is a very old idea. It goes back, well, one can certainly see concrete versions of this as early as the 1940s. What has changed in the last some time is that instead of seeing one neuron at a time, we can now see large numbers of neurons simultaneously. And the result of that is that instead of sort of statistical physics ideas being sort of floating in the abstract, we have the opportunity to confront those ideas with data where we're actually observing large numbers of neurons simultaneously, even numbers which get so large that you start to think that you really need to do statistical mechanics. I noticed that Ken Harris's title is about the number of neurons going to infinity. I won't manage to get to infinity, but we'll pause at 100 and somewhere over 1,000. As many of you know, there are two very different approaches for recording from large numbers of individual neurons simultaneously. One of them is basically to take the old-fashioned idea of putting an electrode near a neuron and picking up the electrical signal and scaling that up. And I think you'll hear about that from Ken, where now you can record from thousands of neurons where you place lots and lots and lots of little wires into the brain, or maybe the modern semiconductor fabricated equivalent of wires. The alternative, which you see illustrated here, is where you genetically engineer an organism such that its neurons produce a protein whose fluorescence is sensitive to the electrical activity of the neuron. The ideal would be to have a protein that inserts itself into the membrane and is directly sensitive to membrane voltage. Those proteins exist. They're not quite ready for prime time. If we have this conversation again in a few years, I'm sure they'll be there. In the meantime, what you can do more straightforwardly is to make proteins whose fluorescence is responsive to the calcium concentration inside neurons, which is a somewhat slowed down version of signal that tracks electrical activity. And in doing this, you turn the problem of recording from neurons into a problem in microscopy. And of course, there have been revolutionary advances in microscopy in the last decades as well. The figure on the left reminds us that you can't just, that there are many challenges to making these experiments work. These particular experiments are being done in the laboratory of my colleague, David Tank. And you're genetically engineering the mouse to produce the right fluorescent protein in its neurons. You've built a state-of-the-art 2-photon microscope so that you can scan and look deep inside the brain with the right trade-offs between scattering and resolution and so on. And finally, most of the things that the brain does, many of the things the brain does, it doesn't do unless the animal is behaving. On the other hand, you would like to hold the animal still so that you can see things at high resolution. I'll remind you that although cells might be 10 microns across, the spaces between cells are much smaller, and so you really need this resolution in order to be able to distinguish the cells from one another. And so this is an experiment which has now become, the style of which has become very popular, in which the animal has its head fixed so that the brain isn't moving around too much, but is otherwise free to move, and it's running on a styrofoam ball, which is suspended on a column of air. The motion of the ball is tracked, as you'll notice, by a different kind of mouse. And that tracking of the ball allows you to determine what path the animal would have taken through space and use that to drive virtual reality for the mouse. So in order to get this experiment to work, you have to do the physics of the microscope right, you have to do the molecular biology of the probes right, and you have to do the computer science of the virtual reality right. It's quite a thing. And so I'm a theorist, I can say that. I have unalloyed admiration for my colleagues being able to do this. So now you can record from lots and lots of neurons. What do you want to do? There's many things you could do, and I want to focus on one notion of what it means to understand. And that notion is that if I look at the activity of the neurons, then I would like to be able to tell you what is the probability distribution out of which the states of the network are drawn. Many different questions you could have asked, some of which are listed here. But in the simple case, imagine every neuron is on or off. So the state of the entire network is an n-bit word. What is the probability distribution over those n-bit words? And just to make an obvious point, but it's worth remembering, you are not going to go out and measure that probability distribution any more than you're going to measure the probability distribution for the positions and velocities of all the gas molecules in this room, because the space over which that distribution is defined is just too big. Okay, so by the time, my favorite version of this is that by the time you're talking about a few hundred neurons, the number of possible states of the system is larger than the number of baryons in the universe. Nobody's going to do an experiment long enough to sample all of those states. And actually, you sort of hope that the brain isn't actually wandering randomly among all of those states. So in order to make any progress, we have to discover that this probability distribution is somehow simpler than it might have been. So how would we get at that? Well, one traditional way of doing this is to say, as a theorist, let me think about a dynamical model for what's going on in the brain and see if I can derive from that distribution, from that model, what distribution the states of the network will come from. And a famous example of this, of course, is the Hopfield model from the early 1980s, where which actually harkens back to some of those ideas in the 1940s about neurons as simple threshold units that add up their inputs, and if it's above if the total is above a threshold, they turn on, otherwise they turn off. And the great step backward, as I think it was described by Gerard Toulouse, that Hopfield made was to consider the case where connections were symmetric, in which case the dynamics have a Lyapunov function, which we can think of as, so that we can think of the dynamics as being like motion on an energy surface. And if you add noise to the dynamics in some reasonable way, you find that the probability distribution becomes the Boltzmann distribution with the Lyapunov function playing the role of the energy. And since this is actually a physics conference, I don't think I need to say more than that. Of course, any dynamical model that you write down is surely wrong in detail. So there's another approach, which is to say you should start with what you can measure and what you can measure reliably from the data. So for example, if you have a reasonably long recording, even from a very large number of neurons, you can certainly measure the mean activity of each individual neuron. If your recording is long enough, and this is a little trickier as the number of neurons becomes large, you can measure correlations between pairs of neurons. If you keep going down this path and start asking me about correlations among triplets and so on, eventually I'm going to run out of data. So let me pause there with just means and correlations. We all know that neurons that have different average levels of activity are probably doing different things. So it makes sense that when we build a description of a network, we should capture the mean activity of each individual neuron. If that's all we say, then we haven't told you anything about whether the neurons are interacting with each other or sharing information or anything else. So for historical reasons, if not, and also from a widely held intuition, you think, well, let's talk about what happens between pairs of neurons. And that matrix of correlations captures something about the interactions in the network, whether those interactions are direct or mediated by variables that you're not watching. And so what you'd like to do is write down a probability distribution for the overall state of the system, that is to say the joint distribution of the activity of all of the neurons, such that the probability distribution you write down agrees with your experimental measurements of the mean activity and the correlations between pairs of neurons. But of course, that's roughly n squared constraints, and we said that there are 2 to the n possible states, so the probability distribution is a list of 2 to the n numbers. They have to add up, they're all positive and they add up to 1. And we now have n squared, or n squared over 2 constraints that they must obey. So how do we close the gap between the roughly n squared things we can measure in the data and the 2 to the n things we would need to know in order to tell you the whole probability distribution? And lots of ideas, but one that my colleagues and I have worked on for some time is to think about the maximum entropy idea, which says that out of all the probability distributions that match the data, I want the one that has the least possible structure in it. And we know from Shannon that the only consistent way of measuring how random something is, how little structure it has, and so on, is to compute the entropy of the underlying probability distribution. And so what we're going to do is we're going to search for the probability distribution that has the largest possible entropy, hence the least structure, but is consistent with the mean activity and pairwise correlations that we measure. And many of you know the answer to this optimization problem, because you know that in thermal equilibrium, you find the probability distribution that has the maximum entropy given the average energy. And so if you go through the calculation that I just described, you get back the Hopfield model, except now the parameters are not things that you necessarily think have dynamical significance, or even things or anything that you pull out of your hat. Rather, the thresholds are things that you have to adjust in order to match the average activity of each individual neuron. And the interactions are something that you have to match, that you have to choose in order to match the correlations between pairs of neurons. And the good news is that once you do this, you've written down a probability distribution for the states of the system, and you're done. Everything you calculate after this is a parameter-free prediction. But there's some things you should worry about. The first one of which has already come up in the questions, so let me answer it. This looks like we're doing equilibrium statistical mechanics. We're not. For example, if the system were in equilibrium, then you would have detailed balance in the transitions among all the states. There's nowhere in this discussion the assumption of detailed balance. We're describing the probability distribution of states that you find at one moment in time. In fact, if you wanted to, you could think about constructing a maximum entropy description for sequences of states. And then manifestly, whether you get detailed balance or not depends on the actual structure of the correlations in the system that you see. For example, if the states that you see always occur 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3 in order, then if you try to match the temporal correlations of states, you will find that the probability distribution you write down actually breaks time reversal invariance, even though it's the maximum entropy distribution. It's the maximum entropy distribution over sequences of states, and so there's no problem. Similarly here, this would be a thermal equilibrium description if the thing in the exponent of the distribution were actually the energy of anything, and of course it's not. Okay. Somewhat more subtle questions are the fact that we've had this discussion as if you measure the expectation values and you measure the correlations, but of course what you really have is a finite sample of data and there are error bars, and so there are worries here. I'd rather not talk about those because they're interesting but sort of technical, and I guess what I will do for the purposes of the next 15 minutes or so is ask you to trust me that we have those under control. There's a deeper question, which is why should models that are simplified in any way work at all? This is, after all, a very complex system, and that is a question I'm going to come back to. But let's, before we have a discussion about whether it should work, whether it shouldn't work, what it means, let's ask whether it does work. And so in order to do that, let me tell you about what we've done. So there's we started doing this in the vertebrate retina with my experimental colleague Michael Berry and several of the people who have passed, students and postdocs who have passed through his group, and a group of theory students and postdocs who have worked with us, all of whom are now off on their own. And this produced a series of papers, the latest of which is now 5 years old. So I'm going to leave that as written and focus instead on our more recent work in the mouse hippocampus, where our experimental colleagues, David Tank and Carlos Brody, have been working. And this is the work, a lot of what I'll be telling you is the work of one theory PhD student, Lenoy Meshulam, actually who recently moved from MIT to the University of Washington, and I should have updated the slide, apologies. So here we're talking about roughly 100 neurons in the hippocampus, and we're going to move up to more than 1,000. And as you'll see, in order to cross that boundary from order 100 to order 1,000, we're going to need a new idea. So let me, I guess since the talk gets recorded, you can always find your way back to these references. So let me tell you a little bit about the example of the hippocampus. So these are indeed the data that you saw before. I'll draw your attention to the fact that when you measure the fluorescence signal from a single cell, so focus in on a single cell, as shown here, you see the fluorescence signal has an enormous signal-to-noise ratio. It's a delight to collaborate with experimentalists who produce data that look like this. It's not easy. There's lots of issues. These data, you can either think of them, you can either then think that the fluorescence signal gives you a continuous measure of the activity of the individual neurons, or if you want, you can discretize. And for the purpose of this discussion, we'll discretize, and then we'll flip back to thinking about the continuous signal. Mostly you discretize in an obvious way because the signal-to-noise ratio is so high. You have these transients, and you can say whether you're in one or not in one. There's a little bit of subtlety that we know something about the dynamics of the molecule that is responding to the calcium signal. And so if you see it coming down but too slowly, that must mean that the neuron turned back on, and so that produces things like this. But this isn't really crucial. If you just said, well, do something obvious to turn this into a discrete on-off signal, you'd be fine. So you go through the prescription, right? You build the maximum entropy model that is consistent with the observed mean activity. So here's the mean activity of each individual neuron. Remember, it's a binary variable, so it's 0 or 1. Of course, most neurons are off most of the time. So these averages are small. If you look at the matrix of correlation coefficients, it's not exactly the covariance matrix, but maybe it's a little bit more intuitive, you notice that there is a peak in the distribution of correlations a little bit to the negative. Large correlations are very rare. When you build the model, what you're essentially doing is translating from mean activities and correlations into effective magnetic fields and pairwise interactions. And so this figure illustrates that transformation. I don't want to make a big deal out of the details here because I don't think that it's especially important for what comes next. As has already been asked in the questions, it is important how you right. If you're going to make discrete variables, you have to slice time. And what we did was to simply choose the video resolution, which is sort of too short because this is a scanning microscope. So there's a natural discrete timescale that is the time it takes to do one scan, which is clearly shorter than the response time of the calcium indicator molecule itself. So we don't really add anything. We're not really doing any violence to the data by discretizing time. In the case of recording directly action potential, the electrical signals of action potentials, the issue of what time scale you choose for discretization is much more important. So, does the model work? So again, remember, once we match the means and correlations, all our predictions are parameter free. We use the correlations between two neurons, right, between pairs of neurons to build the model. So a natural question is, can we predict the correlations among three neurons? You wouldn't want to build a model using the correlations among three neurons because it's not clear that all of those measurements are independent of each other and they have error bars and so on. But you can certainly ask whether your predictions come out right. And if you look, you see this wonderful cloud of data points that surround the diagonal, shown here. But it's a little hard to tell, particularly in the bulk, whether, remember, with 100 neurons, there's roughly 100,000 independent 3-point correlations. So there's actually 100,000 data points on this graph. You can't tell whether they really fit or not. So what we've done is to take a small window of places where we predict a particular 3-point correlation. And then we ask, what is the RMS error of our prediction? That is to say, what's the root mean square difference between what we predict and what we observe? And what is the root mean square error with which you can infer that number from the data? And that's what you see in the middle. And what you see is that those are essentially equal to each other, which means that our predictions of the 3-point correlations are as good as they can be across the bulk of the data. If you did better, you would have been overfitting. And of course, you learn the model from part of the data and predict on another part, so on in all the usual ways. The actual neuroscientists of the audience will remember that the hippocampus is not a randomly chosen region of the brain. This is, for example, the region of the brain in which one finds place cells, these marvelous neurons that are active when the animal is only in a particular physical location in space, or in this case, virtual location, because it's virtual, because he's running in virtual reality. And so you might think, well, I don't know, you inherit correlations from the structure of your place fields, right? The fact that this neuron is active when the mouse is here, and this neuron is active when the mouse is here. If these regions overlap, then they'll be positively correlated. If they don't overlap, they'll be negatively correlated. So maybe if you just take a model in which the neurons are active with a probability that depends on their position, and of course you can estimate what that probability is by looking at the data, then you'll predict all these correlations. And the answer is no, you won't. So if you make that model in which neurons are independent of each other but depend on position, then your predictions for the 3-point correlations are a disaster. It's not only that you don't capture the full dynamic range, but if you look in the bulk of the data, you find that essentially your predictions and reality are uncorrelated with each other. So it is true that half of the cells are place cells, but quantitatively, that observation doesn't get you very far in predicting the correlation structure of the network. If it's really true that if you take this view that I've written down the joint probability distribution and the activity is collective, then if I tell you what most of the neurons in the network are doing, in fact, if I tell you what n-1 neurons in the network are doing, I can predict the probability that that last neuron, the nth neuron, will be active or silent. And the way to think about that in physics terms is that all the other neurons are applying an effective magnetic field to this spin, which is up if the neuron is on and down if the neuron is off. And you can compute what that effective field is by knowing the states of all the other neurons. And then you can accumulate over all the data every moment in time where that effective field had a particular value and ask, what is the probability that the neuron is active? And if the theory is correct, then it should have the simple logistic form that you get from the Boltzmann distribution. And in fact, that's what you see within error bars. But of course, you can also unfold this and ask moment by moment, what does the probability look like? So at each moment you ask, what's the state of all the other neurons? What's the effective field on this neuron? What is my prediction for the probability that the neuron will be active? And strikingly, what you find is that for all the neurons in the network, you predict that most of the time, the predicted probability is essentially 0. And in very small windows of time, you have a little burst of probability where you expect that the neuron will be active. And that is when the neurons are active. And that's true for the place cells, where you might think that somehow it's all working. I mean, although there's no reference to place in this discussion, maybe it's hidden in there somewhere in the matrix J. And so it's not surprising that we get things right. Really, the cells are active because they're in a place, not because you can predict them from the other cells. But it's also true that there is a significant fraction of the cells that are not place cells, and you can see that. You see the same thing even in the non-place cells. Let me just say again, what this is telling you is that the sort of collective, our ability to predict whether neurons are active by thinking about the collective behavior of the network is independent of whether the cells are place cells or non-place cells. And if you try to predict the correlation structure from the place fields alone, it doesn't work. What the animals are doing in this particular experiment is very simple. And of course, you can ask, will we get similarly good agreement between theory and experiment when the animal's doing something else? And that's a good question. There's no claim here that what we're seeing is the result of direct connectivity among the neurons. It's rather a sort of effective description of the collective behavior of the network as a whole. So I hope that gets a little bit at the question that was in the chat.
Bill, for those of you who missed it, could you just, Tommy, just describe again briefly what you mean by a place cell and a non-place cell?
In the hippocampus, you find these remarkable neurons that were discovered by O'Keefe, right? Where if the animal is running along, you'll find that the neuron is essentially silent until it arrives at a particular location in space. And then the neuron is active, and when the animal leaves that region, it goes silent again. And so in this simple, right here, it's happening in virtual reality, but it also works. And that's very important and has a lot to do with how animals navigate and everything else. But for the point of view of this discussion, you might guess that a certain amount of the correlation structure in the network is just inherited from those place fields. So the thing that we tested is to say, if the neurons were active or silent with a probability that depended on where the animal is in the virtual world, then would you get the pattern of correlations right? And the answer is emphatically no, which is pretty interesting. Replacing ensemble averages by time averages is something that I think everybody does in analyzing data, because you don't do the same experiment 1,000 times. What it is equivalent to the assumption that you're in some sort of statistical steady state. And so you have to check that you're not averaging together epochs of the experiment that are significantly different from one another. That's a standard problem for all analyses. And we did our best, as everybody else does. So what I want to do now is to jump. I mean, we were actually quite struck by this level of success. Predicting 100,000 3-point correlations with no free parameters within error bars isn't bad. And in particular, this temporal structure of the predicted activity of the neurons was quite striking to us. And you can tell that the examples I'm showing you are not chosen to be the best ones, because if you collapse all of them and just ask about the overall probability, you get that right as well. But what we really want to do is talk about much larger populations of neurons. And that brings up the question of why you might think that simple models work at all. So let me remind you that it is conventional to describe, for example, magnets by the Ising model, which by now should be familiar. It's the Hopfield model or the maximum entropy model consistent with pairwise interactions. And you know, biologists often accuse physicists of oversimplifying biology. I would say that one response to that we should use more often is to point out that biologists shouldn't feel special. Don't worry, we don't only oversimplify biology, we also oversimplify physics. That is to say, there is no real magnet whose microscopic dynamics is described by the Ising model, the pairwise Ising, nearest neighbor Ising model. That is an oversimplification. Why does it work? Well, you could say, well, it doesn't work. You know, you get that things are ferromagnets and paramagnets and things like that, but you know, it's only qualitative. That's not true. As I think most of you know, in particular, if you ask about the behavior in the neighborhood of the critical point, where fluctuations occur on long length scales, you get things exactly right. You can predict behavior. First of all, you predict the form of the behavior in the neighborhood of the critical point correctly, and you make numerical predictions that are accurate to 3 decimal places and are parameter free, despite the fact that you have a model which is microscopically wrong. So why does this work? And we now know the answer to this. It's not just wishful thinking or good luck. It's that if you think about building a model on a microscopic scale and then averaging over regions of increasingly large size and asking what happens to the model as you change the scale on which you look, the model typically gets simpler. And the number of terms that survive coarse graining from the most microscopic scale out to a macroscopic scale on which you're making your observations is very small. And what that means is that you can get the macroscopic behavior of the system right, even if you only keep a small number of what are called relevant microscopic interactions. And of course, this is the idea of the normalization group. So is it possible that we can use this idea to help us simplify our description of networks of neurons? Well, there's a problem, which is that when we do coarse grain, and all the applications of the normalization group that we're used to in statistical physics, we use length scale, right? We talk about neighborhoods in space. And that's because interactions are local and so on, but neurons are big objects and they interact with each other over distances that cross many neighbors, right? So locality isn't really a very good guide. I mean, neurons in one part of the brain don't directly interact with all neurons in all parts of the brain. So on some scale, locality is useful. But if we're talking about a millimeter square region of the brain, then locality isn't really helping us very much. And so what we're going to do is to use as a surrogate for neighbor the neuron with which I am most correlated. So that works if you really are on a lattice, for example, then it's true that you're most correlated with interactions are local. It's true that you're most correlated with your neighbor, but we're going to use that as the criterion. So what we're going to do is we're going to compute the correlation matrix among all the neurons. We're going to walk through. We take the most correlated pair of neurons and we average those together. We take the next most correlated pair of neurons and we average those together and so on. And you do this until the n neurons that you started with become n/2 superneurons. And now you can do it again and again and again. And this is the analog of what we do in statistical physics as we zoom out in space. So what happens when you do this? Well, actually, there's a question, how do you tell what's happening, right? So instead of having the activity of n neurons, I now have the activity of n/2 or n/4 or n/16 or n/64 superneurons. So what do I look at? Well, one thing you can look at is just what is the probability distribution of that activity? And let's normalize it, right? So that the mean of the non-zero activity is always 1. Well, when you do that, then the probability distribution comes in 2 bits, right? There's the probability that this superneuron is silent, and then there's a distribution of its activity given that it's not silent. And what you see is that as you increase the scale over which you're averaging, the probability distribution of the non-zero activity very quickly approaches a fixed form, which is not Gaussian. So remember that if you take uncorrelated variables and you average them together, or even sufficiently weakly correlated variables and you average them together, the central limit theorem tells you that eventually you're going to get a Gaussian distribution. So what you see here is that although you're averaging together groups of, up to 256 neurons together, you're nowhere near being Gaussian. In fact, you're becoming, if anything, more and more exponential, which is very strongly non-Gaussian. Okay, and that distribution reproduces itself at every scale of averaging. Furthermore, if you look at the probability that the whole group is silent, again, if the neurons were independent of each other, you'd expect that to fall off exponentially on average. Instead, it falls off as a kind of stretched exponential with a non-trivial exponent. And you see that the accuracy of the scaling is quite good. If you try to estimate this exponent from a single experiment, you can measure it to, 1.5% or so. And if you do the experiment on several different animals, you discover that the variation across animals is on the same scale. So if you're a devotee of the normalization group, I hope you understand why these kinds of observations are exciting. If you're not, what I could say is, I've told you about a network level property of this large population of neurons, which we can measure to 2 decimal places, and it is reproducible from animal to animal in that second decimal place, which I find very striking. There are more things you can check. For example, let me not talk about the problem of correlations. Let me just talk about dynamic scaling. So in the example where you're coarse graining in space, you have the intuition that if interactions are local, then for things to change on a longer and longer length scale takes longer. And so, I mean, the simple example of that is diffusion, right? Where the length scale of the time scale and the time scale of changes are tightly linked to each other, right? The time scale is the square of the length scale. More profoundly, if you measure correlation functions, if there is no characteristic scale of length or time, then if we rescale correlations that are measured across different length scales, we should see the same correlation function in time. And what you see is that that's true, even though it's not length here, it's the sort of size of these clusters of correlated neurons that we're talking about. And you see that the accuracy of the collapse of the data is really quite good. And furthermore, the time scale grows with the number of neurons, again, with some simple power, although the power is very small, and so it's not so easy to measure. What this suggests is that this network is not characterized by any characteristic scale of number of neurons, that it's not characterized by any characteristic scale in time, but that rather these can trade against each other over at least the full dynamic range that is accessible to us in the experiments. And maybe that's what's functionally important. So there are things to worry about here. What does this scaling behavior have to do with the fact that there are place cells? Are we screwing something up because, again, the data set's finite and we're now talking about thousands of neurons? Could something like this emerge from a kind of generic network where things are randomly connected to each other? There's a long paper, which is still on the archive, which addresses all of these things. We sort of mentioned them in the short paper. Let me leave those with you. If one of those really bothering you, we can talk about it and sum up. For me, the conclusion from the first part of the work is that these simple models work surprisingly well. Not only might you have expected the opposite just on for general orneriness, there are people who explicitly predicted that these models wouldn't work, so it's always satisfying when they do. There are obviously limits because the strategy that we're talking about breaks down if the number of neurons becomes too large. You can't really measure the full pattern of pairwise correlation. In the same sense, if we do a systematic coarse graining of the activity of neurons, averaging neurons with their most correlated partners, then we see simple orderly behaviors. Let's not commit ourselves to any interpretation of those things, but I think that it's interesting that these are simple. This isn't just dimensionality reduction, because it's telling you that the reduced variables are coming from the same statistical structure as the less reduced variables, which is not what dimensionality reduction means. The behaviors are quantifiable and reproducible. For those of you who come from this part of statistical physics, you know that what we're hoping for is that the behaviors will be universal. That's a little too much to ask at the moment. And this has been entirely phenomenology. Many of you realize that there ought to be a theory underneath it, because if it's really true that the probability distribution is all the statistics are reproducing themselves, except for scaling, as we group neurons together, then there's an underlying theory which in the technical language of the normalization group should be at a fixed point, and we would like to construct that theory. So I hope this has convinced you, or at least given you an introduction to the idea that decades of thinking about statistical physics for networks of neurons is now at a stage where we can confront those ideas with real data on the right scale, and that makes all of this vastly more exciting. This may or may not be the right way to do everything, but I hope to have left you with that sense that it is an exciting time for our field, and that we should demand of our data that it be analyzed in ways that pull out quantities that really have deep quantitative significance in some larger theoretical picture, and on the other hand, demand of our theories that we actually get agreement with data at what I think of as sort of physics level of precision. We've reached the state in these kinds of experiments where we can no longer shrug and say, well, you know, it's biology, so it's a little bit of a mess. I think what you've seen is that it is possible to get to the point of really quantitative agreement between theory and experiment. That doesn't mean the theories are right, but I don't think we're going to go back and let ourselves get away with anything less than that level of quantitative agreement. So let me stop there and thank you for your attention.
Bill, thank you very much indeed. That's created a huge amount of interest, not least in me and in a number of people who've asked questions. And you caught up, you were doing really well with the questions until they started flooding in the last 10 minutes or so. I wonder if I could pick up with modifying just one of them, which is from Pietro Ceccuta, who asks about effectively the effective dimension of your system. So if you're not selecting spatial proximity as your neighbor measure for the renormalization calculation, doing something else, correlation, does that induce a type of dimensionality? Pietro says it seems to be creating a one-dimensional chain, but that depends on.
So I don't think so. Certainly. So maybe I should give them a slight. So I don't think so, but we should. I mean, we should have that. I think there's a real calculation one can do to answer that, and I haven't done it. So let me say I don't think so, but we should have that. Pietro, we should have that conversation.
Brilliant.
Let me make a slightly more general remark, though, which is we chose a coarse graining method. We think it's a sensible coarse graining method, but there are others. And in fact, there are things that are not, you know, why did we group them 2 at a time? We could have grouped them 3 at a time or 4 at a time, which maybe would change the effective dimensionality. We looked for things that were maximal in their pairwise correlations, but you could think about, you know, larger clusters of cells that share maximal information or something like that. There's a whole variety of ways of doing it. We know, of course, that within reason, behaviors aren't supposed to depend on the details of your coarse graining method, but it's also true that some methods are more efficient than others. And in the usual case, your coarse graining methods are highly constrained by symmetries. Here they're not. And so there's much more of an open question of how you should do it. This was a first try.
Yeah. Actually, there's a related question from Viola Priesman, who asks whether the non-Gaussian emergent distribution some of that might come from this choice of
exactly
choosing high
so
correlation.
The worry here is that in effect you're chasing the noise in the correlations, right? I find even if there isn't any structure, I find the 2 neurons that are most correlated with each other, and then the next 2, and so on. And somehow I'll build something. I could even build something out of nothing. For example, if you do a simulation of a randomly interconnected network, then you don't get, then it's Gaussian in this construction. If you do independent neurons that are responding to the position of the animal, so place fields alone, then it becomes Gaussian and so on. So this is, you're right to worry about this, but I think that isn't the origin of what we're seeing.
That's fair. I should just say to everyone, because we started the whole conference just about 5 minutes late, I'm going to add 5 minutes exactly to all the time at the time. So we have our next speaker, Kenneth Harris, already here, but it'll be a time for another couple of quick questions to Bill before I hand over to Ken. Don't worry, Ken, it's nice to see you. By all means, stay visible and join in the discussion on stage here. Bill, I don't know whether you wanted to select any one of these rather nice page of questions we've got that I know you've been looking at.
So let me, hi Rasmus, how are you? Let me try that one. So is there a connection between this and the now common observation that a few dimensions explain most of the variance in the activity of population neurons? There is a connection between PCA and the normalization group. There's actually, for what it is worth, there we go. There's a paper called PCA meets RG. So first of all, in this particular population, it is not true that a small number of principal components explains most of the variance. The spectrum is becomes more and more continuous the more neurons you record from. And so there is no break where you would say it's low dimensional. And it's also true that the normalization group is really about the form of the distribution. If you think about PCA as a way of saying these low variance components are things I'm going to average over and not pay attention to, these high variance things are the things that I think are important that I'm going to keep, then that boundary is somewhat arbitrary. And the normalization group is really about what happens to the variables that are left over as you move that boundary. And the striking thing here is that the distribution of variables that are left over takes on this sort of simple and self-reproducing form as you move the variable. So it's connected, but it's not just that.
Super. Bill, thank you. As the questions we're now tackling are verging into the territory in which I think Kenneth, Carl, and Daniel's contributions might help us answer them.
Yes.
I'm tempted to suggest that we carry on further in the panel discussion at 3 with you, that everyone sort of virtually claps Bill and hoots madly for a fabulously interesting talk. Thank you so much. Bravo, bravo, bravo. And so I'll invite Bill to gently disappear. Ken's here. And Ken, you probably welcome to Physics of Life. If you have some slides to share, then sharing them now. But other than to say,
you see them?
We can. So, number of neurons tends to infinity. Over to you, Ken. I won't. Thank you very much. You're from UCL. I won't waste any more time other than just saying welcome and thank you. Over to you.
Thank you. Thanks very much for the introduction and to the invitation to this fantastic virtual conference. I think there'll be some interesting synergies with the previous talk in what I'm going to say, actually, even at a quantitative level. What I'd like to talk about is some work where we did large-scale recordings in mouse visual cortex using the same method of two-photon microscopy you heard about. We found an unexpected experimental result, a quantitative law in the data. I'm not going to tell you all four of the theorems, just a quick summary of the math that we did, which led to a further experimental prediction and test. So it's an example of what we just heard, this philosophy that you treat biology like physics, making quantitative predictions from theory and then going back and testing them. And this is work done by Carson Stringer and Marius Pacciatario, now at Janelia, when they were in the lab that Matteo Carandini and I run together at UCL. And this is published. It came out last year, and I think this should be open access. But just in case it's not, do email me if you don't have access to the document. Okay, so what I'd like to talk about is the visual cortical population code. So when you see an image with your eyes, that causes a pattern of activity in your brain. And you can imagine you can visualize what that activity might look like in a grid like this. Every point here corresponds to a neuron. The green ones are the ones that might hypothetically respond to this picture of a tree. The orange ones are the ones that might hypothetically respond to this picture of a goldfish. And one of the points that was made way back in neuroscience is that there's a real advantage for these neural codes, meaning these population firing activity patterns, to be orthogonal. So if you have these two representations of these two images, if the vectors are orthogonal, the vectors defined by the firing rates of all these cells, that means the number of cells that respond to both of them is very low. Every cell responds to one or the other, but not both. And that's advantageous because it means these two stimuli can be easily distinguished. On the other hand, sometimes there are sensory stimuli that should cause similar behaviors. So if you see two different pictures of trees, the behavioral response, to climate is the same. So you might want to have a larger amount of overlap between these, which means these vectors would no longer be orthogonal. So we can think about the neural code as a mapping, as a function from a set of stimuli that includes these images. Obviously, it's not really a two-dimensional torus, it's just for illustration, but a mapping from that manifold of possible inputs to a much higher dimensional space, because the number of neurons in the cortex is much larger than the number in the retina. And in particular to a subset or submanifold, potentially a submanifold of that higher dimensional space. So what can we say about the form that this mapping takes? All right, so we're going to analyze this experimentally by recording from 10,000 neurons simultaneously, and the sensory stimuli that we presented while we record in visual cortex are just natural images from the ImageNet database. For a technical reason, we need to present each of them twice. And we use this method of two-photon microscopy you heard about from Bill. This is a movie of the raw data. When you see these cells flashing, that's because they're firing action potentials and calcium comes in. Take an 11-plane scanned image, and you can get around 10,000 neurons simultaneously. The difficult part is not the experiment, it's analyzing the data to extract the cells. And Marius and Carson wrote this excellent program called Sweep2P, which is available freely, and I'd really recommend anyone doing this method to use it. Okay, so here's an example of some of the data. We see a subset of 300 out of 10,000 cells on the x-axis. On the y-axis, you see a subset of 100 or so neurons, and the pixel is red when that neuron responds to that stimulus. So you see this kind of hazy pattern because they've been sorted in order, but certain neurons fire in response to certain stimuli, which means that the neurons encode information about the stimuli. And if we play a game of asking, can we decode what the stimulus was from the neural activity, we find that we can, to actually nearly 100% accuracy, 90% accuracy. And because there are 3,000 images, the chance level is 0.03%. And we're getting close to 90% when you use all 10,000 neurons. And this hasn't saturated either. So the information about the stimuli is absolutely there. The question is, what form does it take? So we use something you heard about earlier, the method of principal component analysis, to estimate the dimensionality. So principal component analysis, if you don't know, it measures the variance of successive dimensions of a set of points. So the size, essentially, of the largest dimension of this manifold would be the first principal component eigenvalue. The size of the second one would be the second, the third, etc. And something I don't have time to talk about here, but it's very important, is that we used a cross-validated version of principal component analysis, because that will focus only on the sensory dimensions, only on the parts of the visual cortical activity that are controlled by the visual stimuli. And we want to ignore everything else, all the noise, all the non-visual related information there. And that's what this cross-validated measure allows us to do. So what we found is something that we certainly weren't expected, which is that if you look here at the cumulative variance of the nth principal component, it's an approximately linear function, right? So the amount of variance up to the first 100 dimensions and then up to the first 1,000 dimensions, when we look at it on this log linear scale, is approximately a straight line. And so if you know that the sum, this means the variance is a logarithm of the total number of dimensions, the cumulative variance. And as you may know, the sum of 1 over n is approximately log n, which means that the variance of the nth dimension alone, not cumulatively, is approximately 1 over n. And this is exactly what you see here in this log-log plot. The variance of the nth dimension is approximately 1 over n. And this is true across all recordings. In fact, it's a power of n that's slightly exponent just a little bit above 1, 1.0304, always just a bit above 1, never below 1. So it's not exactly 1, it's a little bit above it. And this is, first, it's very interesting to hear that, as Bill just told you, they saw similar things in the hippocampus. So it may end up being a general phenomenon, in fact. But at the time, at least, we were very worried about all sorts of potential artifacts that could have caused this. And the simplest way to show that this is a genuine phenomenon is to create a situation where it can't be true, right? So we've got the variance of this cloud. The nth dimension goes like 1 over n. But remember, this is only the variance responding to the sensory stimuli. So if we show not 3,000 stimuli, but just a small number of stimuli, 32 stimuli, we ought to find that this cumulative variance saturates at 32 and then no longer increases anymore, because 32 points always have to lie on a 32-dimensional plane. So that's what we did, is we presented, rather than 3,000 images, we present 32 images, but repeat each of them 96 times, and apply exactly the same analysis method and get this, that the cumulative variance saturates after 32 images. So it's not a technical artifact, it's a real result. What could it mean? Well, the first thing is, could it be something to do with the number of neurons that we record? We actually find that as we record more and more neurons, the range of this power law extends. And if we plot the exponent here in red as a function of the fraction of neurons we consider out of the full 10,000, or as a function of the fraction of stimuli we consider, we see in both cases, as you increase the number of neurons and stimuli, it converges to this value just a little bit above 1. Okay, one other potential explanation for this, because we're looking at the visual system, as you may know, visual images themselves have a 1 over n power spectrum. So if we play the same game, do the same analysis on now the image pixels, rather than the neural responses to the images, you see also something quite close to a power law with an exponent just above 1. Could that be the basis of the neural power law? Well, the answer is no, because if we whiten the image to break the power law in the image pixels, it's still there in the neural data. Okay, so that's a lot of things that do not explain the power law. What might be the functional role? Why might we see this in the data? So what we've found is that as the number of neurons tends to infinity and the number of stimuli tends to infinity, the eigenspectrum, the PCA spectrum, gets closer and closer to this limit of a power law just above 1 over n to the minus 1.03 or so. What does this tell us? And this is where we bring in the mathematics. So first, what does it even mean to say that the number of neurons tends to infinity? There's a finite number of neurons in the brain. What are we even talking about? Well, you do this sort of thing anytime you use statistics. Suppose we said, the firing rates of these neurons that we measured follow a log normal distribution. That's something you hear a lot because it's a fairly good approximation. Is this statement true? No, taken literally, it's not true. There's a finite number of neurons. They must follow a discrete distribution. What it really means is that these neurons look like a sample from an hypothetical infinite population of neurons we might have recorded, and only that infinite population can follow actually a continuous distribution like the log normal. What we're saying is our data look like a random sample from an infinite population. So in our case now, we're doing something just a bit different to that, which in statistics goes by the name of functional data analysis. We're considering that the stimuli we actually showed were drawn from a probability distribution of stimuli we might have showed, and the neurons that we recorded are drawn from a probability distribution of neurons we might have recorded, with the understanding that this isn't just the neurons in that brain, or even in all the brains in the world, it's the distribution of neurons that that brain might have contained in principle. So the point is that we can summarize then this infinite population code as the noise-free response of any potential cell to any potential stimulus. These are now infinite dimensional spaces. Of course, we can't measure this function, but we can hypothesize that the actual responses look like a sample from these distributions, and we can ask, what do we infer about this infinite dimensional, infinite population code from our finite sample? Okay, and I'm afraid I don't have time to get into the details, but the math is basically as follows. In this functional data analysis framework, you can consider the neural code as a mapping from the stimulus manifold into an infinite dimensional Hilbert space, which is the space of all square integrable functions on the set of cells that might have existed. So in other words, we have this space of all possible cells and a square integrable function on that set of all possible cells is the representation of a particular stimulus in this Hilbert space. And that is the infinite dimensional code that we can't measure, but we can still infer something about. And what we're able to prove is two facts. So if we take the PCA spectrum of the image of F, we can show that if that spectrum decays slower than n to the -1, then F, the mapping from stimulus space into this Hilbert space of neural activity can't be continuous. And if, furthermore, it decays with a weaker condition at a rate of n to the -1 minus 2 over d, where d is the dimensionality of this stimulus manifold S, then F it may be continuous, but it cannot be differentiable. And in particular, that means the neural manifold, the space of possible activity patterns within the high dimensional space, within the Hilbert space, has to be a fractal. Okay, so let me give you an illustration of this. What we're seeing here, it's a two-dimensional torus that's mapped into a 10,000 dimensional space with an eigenspectrum constructed to be a power law. And then we look at three random projections of it to visualize that in three-dimensional space. Here's the stereo view, and here we see the three coordinate functions. And we're animating so that the power exponent of this eigenspectrum is getting closer to 1. And now we're here, just about to cross the border 2, which 1 plus 2 over d, when d is 2, that is the point at which this manifold became a fractal, no longer a differentiable manifold. It's starting to look rougher and rougher in the fine details. And when we cross the exponent of 1, now it's no longer even a manifold, because this map is not continuous. It starts to look like a cubist painting. The point is that any 2 points here, however close they are together, will be mapped to very different parts of this neural activity space, because this map is no longer continuous. Okay, so this is a piece of pure mathematics. We were able to prove, whoops, let me say it again, prove these two things. But the exciting part is that we can use this mathematics to make an experimental prediction. Okay, so what the math tells us is that if we have a decay of eigenvalues faster than n to the -1, it's not continuous. The neural code can't be continuous. That's a disastrous neural code, it's absolutely useless. If it decays a bit more faster than that, but still slower than n to the -1 minus 2 over d, where d is the dimensionality of the stimulus manifold, then the neural code cannot be differentiable. If d is large, as it is for the natural image database we start with, then 1 plus 2 over d is around 1, so we can't tell the difference. But if d is smaller, we can distinguish them. So what we did is to create stimuli of a lower manifold dimension and ask, does the eigenspectrum now decay faster? Because the theory is that any sort of non-differentiable neural code is no good. It's not something you want. So the brain will organize itself so that the eigenspectrum has to decay faster than n to the -1 minus 2 over d. And that's a stronger thing than we just saw. We just saw it decay slower than, faster than n to the -1. But when we have a lower dimensional stimulus set, we should have an even slower decay than that. So what we do is we create low dimensional visual stimuli. So take the original images we showed, project them onto a set of basis functions, here 8 basis functions, and create these 8D, this database of 8D images by projecting the original ones onto these 8 static basis functions. Okay, and 1 plus 2 over d, if d is 8, is 1.25. So before we got these power law spectra with exponents of, say, 1.03, that will no longer cut it if the hypothesis is true. That's less than 1.25. It needs to be greater than 1.25. What we actually observe is, again, we get a power law, but now the exponent is closer than 1.5. It's on the right side of that value of 1.25. If we do a 4-dimensional image ensemble, then now the critical value is 1.5. So this isn't any good anymore. It needs to be larger than 1.5. And that's, again, what we see, a power law, but with a larger exponent again. And in fact, over a range of stimuli, you see that the decay of the eigenspectrum is close to the n to the -1 minus 2 over d bound, but always just on the right side of it. So to summarize, theory and mathematics tells us if the eigenspectrum decayed slower than n to the -1 minus 2 over d, then the population code couldn't be differentiable. If it decayed even worse, slower than n to the -1, it couldn't even be continuous. Experiment tells us it's faster than both, but close to n to the -1 minus 2 over d. What that means is that the brain is giving almost as much weight to the fine details of the stimulus encoded by these small principal components as it can afford to before these dominate the representation. So the interpretation of this is that neural codes have to balance 2 desirable properties. First, orthogonality, so different stimuli can be easily distinguished. And slow eigenspectrum decay means more orthogonality. Second, smoothness, so similar stimuli can be represented similarly. And faster eigenspectrum decay means more smoothness. It decays just fast enough to the code to remain smooth. And one last slide I want to show is that there's some resonance of this to something you see in artificial neural networks. So deep neural networks, as we know, are fantastic for image recognition, but they have this flaw, which is that you can take an image and change just a few pixels by a tiny amount and completely confuse the network. So this originally was classified as a panda, but after this small noise, now appears to the network to be a gibbon. This reflects a lack of smoothness because the derivatives are too large. It's no longer differentiable. And it seems the brain may have found a way around that problem. Okay, so thank you very much for your attention. These are the people that did the work, are very generous funders. And again, I'm happy to send a paper if anyone doesn't have access. Thank you.
Kenneth, thank you very much indeed. More extraordinary behavior emerging at large numbers and a sort of renormalization thing. This is beautifully following on from Bill's. Thank you. There are a couple of questions in the chat, but in the interest of particularly important for web conferences, sticking bang on time, and we're T+5 minutes which is how because we started 5. I want to keep with that. So Ken, if we can handle those questions either offline as Bill did for many of his, or in the panel, hang around for the panel. I shall immediately, if you just end your share for the moment. But we'll hang out. We're going to stay with UCL, however, because our next speaker in this section on perception, and I think is Carl Friston. Carl, thank you very much for joining us. And I'll just hand straight over to you. Many thanks.
Thank you very much. It's a great pleasure to be here. My head is still spinning after Bill and Ken's talk, so I forgot what I was talking about, but I'm now remembering. So I'm going to bring you a question that has preoccupied me for about 2 to 3 years now, and I haven't got an answer for it. It's very abstract. I think it's very important. I'm not sure whether you will think it's very important. It's basically a missing link that would connect objective functions in things like machine learning or the Bayesian brain hypothesis to the physics and indeed the ensemble dynamics that we've heard so beautifully dissected in the previous 2 talks. So it is a question, it's an unanswered question. There's one slide at the end. So my talk is just basically setting up why this question is important. And the trick, or what I'm going to do, is basically say, we think we know what the ultimate objective function is for all sentient behavior, for a mechanics of self-organization of living things, including sort of mice and men. And in particular, picking up a theme which we've already heard twice now, that it's not just the understand that behavior in terms of probability distributions and ensemble dynamics, but it is the behavior of the system itself, which we also have to account for. So I'm going to tell a quick story where to convince you that we might know the functional form of this objective function, and then say, well, where did that come from? And if it came from physics, from stochastic thermodynamics, and in particular integral fluctuation theorems, then there's a missing link. So that's what I'm going to do. I'm going to talk about sort of self-evidencing. So I'm going to just start by saying, if it is the case that the objective function that accounts for everything, whether you're doing deep learning or whether you're doing cognitive neuroscience or certainly computational neuroscience, can be cast as a bound on the evidence for your models of how the world works, then you'd normally commit to optimizing a free energy functional of beliefs or approximate posterior beliefs about the causes of your sensations or states of the world, latent states of the world, generating observations. So this is a fairly standard objective function that you'll find in high-end deep learning, such as variational autoencoders, where it's known as an evidence lower bound. And it's basically the evidence, the probability of some observations or data that we have to contend with under a given model of how those data were caused, bounded by something which is never negative. Hence, if we now optimize or extremize this functional, we can associate it with evidence. It's nice if you were doing artificial intelligence, because the whole thing is predicated on a generative model, which means you can explain your beliefs about how these data were generated. It can lead via optimal Bayesian design principles to the right kind of data mining, and therefore accounts for things like exploration and exploitation, epistemics, and in machine learning, sort of data foraging and data mining. If you're a statistician just analyzing data, then this is mathematically the same as finding the simplest explanation for some data that is accurate. So you're minimizing the complexity whilst maximizing the accuracy. So this free energy functional is just the difference between the accuracy and the complexity. That's just a statement of Occam's principle. It also can be unpacked in terms of minimizing the computational complexity, and you get into takes on this optimization from the point of view of algorithmic complexity. And indeed, if you're an engineer designing computers that run on the minimum amount of electricity, you could appeal to Jasinski's inequality and Landauer's principle. So from everywhere you look, this free energy functional seems to play a key role in dictating what is a good way to make sense of data. I'm going to suggest that, and license that suggestion with a few examples, that this also works in terms of what to do next, in terms of making decisions, behaving, planning. And in essence, planning, the good plans are those that optimize or extremize this free energy functional in the future. An expectation over some posterior predictive beliefs about what would happen if I did that. Interestingly, as we'll see in a second, this decomposition of this functional into accuracy and complexity sort of pops up as, or becomes, two imperatives that underlie Bayes optimality, but in very different flavors. So the accuracy now becomes an expected accuracy of the kind that resolves ambiguity, underwrites long-standing objective functions for optimal Bayesian design, experimental design, while the complexity, the expected complexity, becomes risk, and that underwrites optimal decision making. So that's how I would motivate it if I was talking to people in say, deep learning. If I was talking to students in cognitive neuroscience, I'd ask them this question: what are you going to do? What's the best kind of behavior? Imagine you're an owl and you're hungry. What are you going to do? Well, you're going to search. That's the answer they normally give, and they would be absolutely correct. You're going to resolve uncertainty about where your prey is, and once you've done that, you can predate that prey. And I use that example just to highlight two different ways that you could formulate a functional or a function that describes the way systems self-organize and adapt and plan and behave. So you could either choose to do what people in engineering and reinforcement learning do, which is to assume some state value function of states of the world that would ensue following an action upon those states, and then maximize that value function. Or what I'm going to suggest is the more appropriate way to do it is to use a functional, and it's going to be an expected free energy of a probability distribution or a belief about what would happen if I did that. That has the added advantage that you can do sequential policy optimization and talk about the goodness of sequences of actions into the future. I like that formulation because it really highlights the difference between using functions as opposed to functionals and energy functionals as the basis of the principles of sentient behavior, or physics of sentience, in the sense that if we're looking at the path integral of an energy functional, then we're just talking about Hamilton's principle of stationary action. And that's in contrast to Bellman's optimality principle. And I'm going to try and convince you that this is the right functional form, and that all behavior can essentially be reduced to a principle of state, a variational principle of stationary action. So here is this notion of choosing things to do, acting in a way, behaving in a way that optimizes an expected free energy. That is just the expected evidence law bound in machine learning under a posterior belief about what would happen if I did pursue a policy here, pi. So here's the overall, if you like, functional architecture of action perception or exchange with a heat bath or some external milieu. We take the outcomes, we compute a belief by optimizing the free energy itself, the evidence law bound itself, to form posterior beliefs about the outside world. And then we use those to run out into the future and evaluate the expected free energy, sample from the beliefs about our policies to provide an action. We perturb states of the world out there, they generate new observations, and so the cycle continues. So in trying to convince you, and possibly me, that this is the right functional form, I'm just going to establish its constant validity in terms of the kinds of objective functions you see and abound in different corners of the life sciences, from economics through to behavioral psychology. So what I've done here is written out the free energy here in alternative forms as a mixture of complexity and accuracy. So this is the statistician's view of good ways of updating Bayesian beliefs, approximate Bayesian beliefs about the states that generated my data. I want to maintain their accuracy, but I want to do it in a simpler way as possible with inducing the minimum complexity cost. And this is equivalent to finding the maximizing a bound on the marginal likelihood or the evidence, and bringing my approximate Bayesian or posterior beliefs close to the true posterior distribution over states given outcomes. Here is exactly the same function, but just taking an expectation over the outcomes in the future, which now become random variables. Complexity becomes risk, accuracy or inaccuracy becomes ambiguity, and I can also rearrange this expression where the evidence bound becomes an intrinsic value and the log evidence or log marginal likelihood becomes an extrinsic value. So why have I labeled it like that? Well, just as a nod to other ways of articulating or understanding the imperatives inherent in this expected free energy. So if we forget about the extrinsic value for the moment and just look at what this term entails, this is a KL divergence between beliefs about future states of the world if I did that, relative to my beliefs if I could actually see the outcomes. So if effectively it's the information gain. So if I rewrite that as an expected KR divergence, what I've essentially done is written down an expression, an objective for good epistemic foraging, for epistemic affordance that in the visual search neurosciences is known as Bayesian surprise. The KL between the posterior and a prior that is and is not informed by some observations. And this is just the expected mutual information between the sensory outcomes and their causes, given a particular way of moving through the world or under a particular plan or policy. If I now withdraw a certain kind of uncertainty from the table, and pretend that I had direct access to the states generating my observations, which is quite a common assumption, a simplifying assumption in many aspects of psychology and in economics, then I'm basically left with risk-sensitive control because I've taken away the ambiguity. So states become outcomes. And this just leaves me with a KR divergence between what I anticipate will happen under a given plan relative to what a priori I prefer or expect to happen. And this, I repeat, is known as KL control in engineering or economics risk-sensitive control. And if I take away the final kind of uncertainty, so I'm now removing both risk and the ambiguity, I'm just left with this extrinsic term here, which I can now interpret as in terms of the expected utility, the kinds of things that I expect to encounter in my lived world. And this, of course, relates directly to things like reinforcement learning and utility theory in economics. So in summary, what we're saying is that this functional form, this expected free energy is just expected value under some prior preferences about outcomes, just for Bill's, not to Bill's talk. So this, if you were taking a physicist's perspective on this, this is a specification of the non-equilibrium steady state that you would aspire to. If you're an economist, it would be the expected value, plus the information gain. So this is the Bayesian optimal design, this is the exploration, this is the exploitation. But they're both just two ways of decomposing, or two parts of a carving of the expected free energy that is just the mutual information plus the expected utility. I won't go through this in any detail, but you can unpack this imperative, this expected free energy functional, and show that it can be realized in a biologically plausible way using little rats moving around in their mazes or on their little air balls. This is an illustration where we've modeled the behavior of a rat that can is searching for a reward in one of these two arms of a T-maze. It doesn't know which arm the reward will be located, but it can choose to use one of its two moves to go and look at an instructional cue that tells it where it is and then secure the reward. So it can either take a gamble and see whether the reward is on the left or the right, or it can use up one of its two moves to go and check where the reward is and then go there. Expected utility is exactly the same in both instances, but of course the information gain is much greater if it has this explorative behavior that is then subsequently followed up by an exploitative behavior. And we can formalize that in terms of this free energy functional, because we can write down a simple generative model, in this instance a Markov decision process, that enables us to compute and then optimize using off-the-shelf what's called variational Bayes or variational message passing, that looks very much like how the brain works. Again, this is not the time to go into it, but it always impresses me how just writing down a generative model, writing down the free energy gradient flows and solutions to the underlying fixed points produces updates, belief updates that look very much like the same equations you'd use in simplified computational neuroscience in terms of belief updating, reward prediction errors, softmax, classical softmax decision rules, associative plasticity in terms of learning and action selection. So here's just an illustration of the behavior when we play a trick on the rat or the mouse, where we leave the we alternate the position of the rewards for the first few trials and then leave the reward on the same side, in the fond hope that if everything is working, there's going to be an epistemic affordance. There's going to be this sort of intrinsic motivation to choose policies that go and resolve uncertainty, to minimize this expected free energy, until there is no more uncertainty to resolve, until the rat becomes, or the mouse becomes, familiar with its environment, and then the only imperative in play then is this extrinsic value or the utility of exploiting the now known position of the reward, which is on the left arm here. I think that's exactly what you see after about 20 moves, just by selecting policies that are scored, if you like, in terms of the expected free energy. So here's the question. This is the last or the penultimate slide. If it's the case that all working systems, sentient systems, from mice to men, can be cast as choosing policies or paths through the future that optimize this expected free energy here, which I've in this instance written down in terms of risk and ambiguity, can that be linked to physics? So why would it have this particular functional form here? And by physics here, I'm going to assume that all of physics, certainly all of physics that can be cast in terms of stochastic thermodynamics and ensemble densities, can be written down in terms of a fluctuation theorem, so following people like Seifert in terms of a master fluctuation theorem, and in particular an integral fluctuation theorem. I'm assuming that if you can describe any world in terms of an integral fluctuation theorem, you've connected with physics, and we know that this describes how the world works, or variations on this form of integral fluctuation theorem. So in this instance, I've just written down how the world must work just by writing down a probability distribution, which is this non-equilibrium steady state distribution in the very distant future over these three random variables: the outcomes, the states that are causing them, and the policies that we pursue. And then the posterior belief, as if I had been prepared at the beginning now in some particular probabilistic state, and then rolling out into the future. So as t goes to infinity, this should actually converge to 1, and the log will converge to 0. And there are all sorts of different interpretations. This would generalize the second law if we were talking about equilibria, but we're not. We're talking about non-equilibria in this instance. So how do I get from this to this? And the answer is, I have to assume this is true. I have to make this one small move here. If I can assume that the expected log probability of the kinds of policies, of paths that I pursue given a particular state is equal effectively to the conditional uncertainty, or sorry, the conditional certainty or the negentropy of observations given their causes. If I can assume this is true, if I substitute this equality here into this integral fluctuation theorem here, then it is straightforward to show that the expected free energy does indeed provide a bound on the probability of pursuing a particular policy, and in the limit that those policies roll out sufficiently far into the future, there is an equality. So my question to you, and one that I've been trying to answer for, I repeat, a year or so now, is where on earth does that come from? Involving three random variables. Again, a little nod to Bill's talk, that it's a bit more subtle than mutual information. There's something else going on here that relates causes and consequences and our behavioral or enacted intervention between the mapping between causes and consequences as we sense them. So I will finish now, giving the last word to Einstein, reminding you that evidence is simplicity plus accuracy. Everything should be made as simple as possible, but not simpler. So thank you very much for your attention. These are all the people whose ideas I've been talking about.
Well, thank you, Carl, very much indeed. Consider a 220 pairs of hands in rapturous applause after that tour de force. Thank you. Maybe a few questions in the chat coming up, but in the interest of time, what I'll do is suggest that you have a look at them along as Ken to, and Bill did after his talk. And what we'll do, we'll move straight on to Daniel's talk, and then take questions together in the panel discussion in 20 minutes time. So it gives me huge pleasure to introduce my colleague from York, from psychology, Daniel Baker, who's going to be talking to us about algorithms in neural signal computation. Daniel, over to you.
Thank you very much. Okay, so I'm going to talk today about how the brain combines signals, and I have two overarching objectives, really. The first is to try and build computational models that allow us to link people's perceptual experience with activity that's happening in their brain. And the second is to see if these models reveal fundamental principles of neural function, fundamental truths about how the brain works. So today, I'm going to talk about a particular set of studies that I've been involved in over the past 15 years or so. We'll start off by briefly talking about Erwin Schrödinger, the physicist. And because Schrödinger is very closely associated with cats in the popular imagination, I've got one image of a cat, and that's the only cat I'm going to show today. He's called Dobby. After we talked about Schrödinger, we'll go through some psychophysical and EEG and fMRI results on binocular combination, so how the brain combines signals across the eyes. We'll then look at how the brain combines signals across space and across the ears, and then we'll talk about sort of general themes that come out of all of this work. Okay, so it's perhaps not particularly widely known. It's not something I knew until I started working in this area, but back in the 1920s, Schrödinger took a break from physics, what he described as a break from the difficulties of fundamental physics. And he turned his attention instead to trying to understand how the brain combines signals across the eyes. Now, we'll talk in a moment more about the paradigm that he was working on. But Schrödinger proposed an equation, and that's going to be a very influential equation throughout my talk today. It's an equation that shows how we combine signals from the left and the right eyes, denoted by L and R here. And the critical feature of this equation is that there's a cross-channel suppression term, in the sense that the signal from the right eye inhibits the left eye because it appears on the denominator of that left part of the equation. And similarly, the signal from the left eye inhibits the right eye. It appears on the denominator of the right-hand part of the equation. We can draw that as a sort of diagram, as I've done up the top here, where we have two channels, L and R, and those orange arrows just show the inhibition between the two channels. So the suppression, that's what's known, at least in sort of neuroscience terms, as a gain control. So you can think of it like a stereo. We're adjusting the volume, the response of one channel based on how much activity there is in the other channel. And this equation, which we can trace back to Schrodinger's work in the 20s, turns out to be broadly applicable to how the brain combines sensory information. We'll go through several examples of that throughout today's talk. Okay, so Schrodinger was working on a paradigm in which participants are shown one luminous disk, a circle, and the brightness of that disk is dependent on the amount of light that's shown to each eye. We can change how much light each eye receives experimentally, and we can have our participants adjust the total luminance that they're receiving until the perceived brightness of that stimulus matches some standard, and the standard usually has equal luminance in the two eyes. We can plot the data that comes out of this sort of experiment in the way that's shown in panel B there. Each point there is a perceived brightness for a different ratio of left and right eye luminances. What we can do when we plot the data in that form is compare the empirical results to some intuitive models. So the negative diagonal there that's marked linear, the dotted line, that shows what would happen if the brain was linearly combining the signals from the left and right eyes, just adding them together. In that situation, you would need twice as much luminance if you show the stimulus to one eye as when you show it to both eyes. On the other hand, the dashed line that forms a box marked max there, that shows what would happen if the brain instead selected the brightest stimulus and based its perception of brightness just on that. And you can see that the data points fall relatively close to the linear prediction over part of the range, but at the extremes, they kick back round towards the max operator. Now, those results are very well captured by the equation that Schrodinger proposed, or a generalized form of it at least, where we have each of those left and right eye signals being raised to some exponent. And by adjusting the value of that exponent, we can change the model behavior. So for small values of the exponent, we're quite close to the linear prediction over much of the range. And for larger values, we tend towards the max prediction. So this particular equation does very, very well indeed at explaining those binocular brightness matching data. And in other work, including a paper of my own, we've extended that to look at perception of contrast. So if we have sinusoidal modulations of luminance, that produces contrast signals, which the visual brain is particularly excited by. And if we do the same sorts of things, binocular contrast matching, then we're also able to explain this very well with Schrodinger's equation. Now, I'm aware that physicists refer to Schrodinger, a different equation as Schrodinger's equation, but in my world, Schrodinger's equation is this thing. Okay, so that was very interesting. And I got particularly engaged with this because about 15 years ago, I was doing some work on binocular contrast discrimination. Now, contrast discrimination is a different task from the brightness matching, whereas the matching measures your perceptual experience. Contrast discrimination is a performance task. So there we look at people's threshold ability for detecting a small increment of contrast. We do that relative to some background. So we will present some pedestal contrast, and then we will present the same stimulus with a small increase in contrast. And we ask, how much do we need to increase the contrast before people can detect that it's different? So that's a performance task. And we can do similar sorts of arrangements. So we can present the stimuli to both eyes, binocular presentation, just to one eye, a monocular presentation. And then we can also show the pedestal to one eye and our target stimulus to the other eye. That's called a dichoptic arrangement. So the eyes see different stimuli. What we do in this paradigm is we measure the detection threshold as a function of the pedestal contrast. And we measure these characteristic, what are called dipper functions, where performance first improves over low levels of pedestal contrast, and then our performance gets worse, our thresholds increase at higher levels. And we get this characteristic pattern of monocular, binocular, and dicoptic dipper functions for these different ocular arrangements of the stimuli. And we were able to describe these data using a very similar equation to the one proposed by Schrödinger back in the 1920s. So this variant of the equation adds an additional term, that's saturation constant S. That's just a constant that allows us to set a threshold down at low pedestal contrast. So it's a sort of incidental thing to add into the equation there. But the critical thing is that we still have this mutual inhibition between our left and right eye channels that occurs before the two eyes signals are summed together. So we have suppression before binocular summation. And that model architecture, which has its roots back in Schrödinger's equation from the 1920s, has been successful in describing a whole range of binocular phenomena, starting in 2006 with a paper in Journal of Vision, but also many subsequent studies. So that's perception. And I said that one of my aims was to try and link perception and neural response using a computational model. So the next target then is to see if this model is able to predict the responses of our brains to different binocular stimuli. The model that I've been describing makes a very interesting prediction, which is quite unique, in fact, in terms of other ways that we could arrange those left and right channels and nonlinear transducers and so on. The prediction shown in the bottom right graph here, and it is quite simply that at high contrasts, monocular and binocular responses, so responses to stimuli shown to one or to two eyes, should be approximately equal. And if you think about it, that's consistent with our perceptual experience. If you open and close one eye, the world doesn't really change that much in terms of its luminance or its contrast. It looks broadly similar. But alternative models, and I've shown 5 examples here, do not predict this. So what we're looking at here is the grey dashed line and the black solid line are pretty much on top of each other in that bottom right panel, but they diverge quite substantially in all of these other 5 panels, which show different possible arrangements. For example, what would happen if we summed the signals across the 2 channels first and then put them through a nonlinear transducer, or what if we had 2 separate nonlinear transducers that don't inhibit each other, so on and so on. So this very specific prediction is called ocularity invariance, and we can test this by measuring the brain's response to different ocular arrangements of stimuli. So the first experiment that I'm going to describe that we did this in used a method called steady-state EEG. And this is something that some of you might be familiar with, but I'll describe it anyway for those of you who aren't. It's a great one if you come from a sort of physics background. It's a really intuitive and exciting method. The idea is that we take a stimulus, for example, a grating patch, and we flicker it on and off at a particular frequency. In this experiment, we flickered it on and off at 5 Hertz, so 5 times per second. And what that does, that flicker entrains the firing of populations of neurons in the visual parts of the brain that are responsive to that stimulus. So if we measure brain activity non-invasively by placing electrodes on the scalp using an EEG system, measuring the electroencephalogram, then we can take that waveform, that EEG signal, we can take its Fourier transform, and what we see in the Fourier transform is a very clean, very well isolated signal at exactly the flicker frequency that we're modulating our stimuli at, so at exactly 5 Hertz. And this is localized to the back of the head, so it appears over posterior electrodes above early visual cortex. And lo and behold, when we do this for monocular and binocular presentation, up at the high contrasts, we see very similar amounts of response in our 2 ocular arrangements. We published this in a paper in Cerebral Cortex back in 2017. That was myself and Alex Wade, who you'll hear from tomorrow. And it's worth mentioning that the model makes a whole range of predictions, which we were also able to test quantitatively. So I'm highlighting the ocularity invariance effect because it's a straightforward one to explain, but there's a whole pattern of about 25 distinct conditions that the model is able to reproduce and predict, including situations where we present stimuli to the two eyes that flicker at different frequencies. We also see the ocularity invariance effect when we measure brain activity instead using functional magnetic resonance imaging. Now, the first people to show this were Moradi and Higa in a paper in Journal of Vision in 2009. They measured fMRI responses in primary visual cortex in V1, which is the first place in the brain where signals are combined across the two eyes. And they compared responses to monocular and binocular gratings, which are shown there, indicated by the lower of those two yellow arrows, and also monocular and dichoptic plaids, shown by the upper arrow. And you can see that each of those pairs of response functions there as a function of time are very close together. So we again see that ocularity invariance effect when measured using fMRI, and work in my own lab over the past 2 or 3 years has replicated this finding, again using fMRI, but using different stimuli. So this is looking pretty good so far. We've managed to come up with a fairly simple model which links not just, which explains not just people's perception, but also what's happening inside of their brain, measured in 2 different ways. But we can go beyond this, because it turns out that this model arrangement isn't limited to just looking at how signals are combined across the eyes. Back in 2007, 2 colleagues of mine at Aston University, Tim Mees and Rob Summers, realized that we could extend this general approach from combining signals across the eyes to combining signals across space. So instead of the left and right eyes, we can look at adjacent spatial locations. And we can study this by taking an extensive stimuli, like this grating on the left here, and removing parts of it using a smooth plaid modulator so that we end up with a bunch of holes in the stimulus. We call this a Swiss cheese stimulus because it's full of holes. And so we can think of these stimuli as being analogous to monocular in the cheese case and binocular in the full field grating case stimuli. So they're like the spatial analog of isolating stimuli across the two eyes. And we see that when we run the same sorts of Dipper experiments as I described before, this time comparing checks and full stimuli, then we again get very similar responses, very similar results to what that model predicts. And if we then measure steady state EEG responses, this time using a variant that's called the Battenberg stimulus, here we have little individual microelements and we remove half of those in a kind of checkerboard arrangement. When we use the Battenberg stimulus, we again see very, very similar responses for the full and the checked stimuli. So those grey squares and black circles there are almost on top of each other, and particularly up at the high contrasts, just as that model predicts. Now that on the face of it is a very counterintuitive and surprising result. If you look at what's happening there, we've got 2 stimuli which produce the same response in the brain, yet one of them has twice as much stuff in it as the other one. One of them has twice as much contrast, twice as many elements as the other. The reason that the model behaves this way, and presumably the reason the brain behaves this way, is that we have mutual inhibition, mutual suppression between those 2 spatial locations. So we have lateral inhibition there, which is balancing almost exactly the additional drive when we add in those extra elements. So that's signal combination over space. And then we also have extended this beyond vision and looked at the same kind of paradigm for hearing. So here we've looked at signal combination across the ears, so binaural signal combination. To do this, we create a stimulus where we have a carrier wave, a 1 kilohertz tone, and we modulate that sinusoidally at a lower frequency, so 40 hertz in this example. And then again, we can do the same trick. We can present the modulated tone to one ear, a monaural condition, to both ears, a binaural condition. And we can again measure increment detection thresholds to measure Dipper functions, and we can measure steady state EEG responses directly from the scalp. I'm kind of running out of time here, so I'm just going to say that, well, basically the lines are right on top of the points, and the model here is exactly the same architecture. The only change for hearing is that it turns out the suppression between the ears using this paradigm is much weaker than suppression between the eyes. So going back to our model, we can add in a weight term on that cross-channel suppression on the denominator. The weight for vision is 1, and that's why we've not mentioned it before, but the weight for hearing is much weaker. It's less than 0.2. Okay, so my last substantive slide, I can see Tom's appeared, so usher me off the stage.
Couple of minutes if we can. Great.
And my last substantive slide, we're asking why the brain does this. So it seems that we have a kind of general approach to signal combination that works pretty ubiquitously across all of the different stimulus dimensions that we've tried it on. Alex Wade and I noticed that this equation that we can rewrite slightly by adding weight terms to all of our signals there is more or less equivalent to what's called a Kalman filter. Now, Kalman filter is widely used, particularly in engineering. It's the optimal method for combining two noisy signals. And in neuroscience, it's been previously used to explain how signals are combined between cues, so across the senses, between, for example, vision and touch or something like that. The idea of the Kalman filter is that you downweight noisier signals. And what I think might be going on here is that that gain control suppression on the denominator of the equations there, that might be the method, the mechanism rather, by which the brain implements the optimal signal combination that happens in the Kalman filter. Of course, most of the time in, for example, binocular vision, we anticipate that the two eyes have approximately equal noise, they're equally noisy, and so the weight of inhibition should be balanced between them, as it is in our general model. But if we extend this to multiple cues, as many people have done in the past, then we might expect those weights to become unbalanced. So we started with a very simple equation that Schrödinger proposed, and we've seen how this general scheme for combining signals accounts for a very substantial breadth of results from perceptual judgments through to neural activity. And I think that this tells us that perhaps the purpose of this gain control suppression, which is ubiquitous in neuroscience, and there are many dozens of papers that explaining how this works, the purpose of it could be to implement some form of statistically optimal signal combination across multiple inputs. I'd just like to say thank you to various people involved in these projects, both at Aston University and here at York, and various organizations that funded this work. And also thank you for listening.
Daniel, thank you so much. And again, round of applause virtually. That's just fascinating. I have a bunch of questions, but I think what we'll do is situate questions together with with a discussion with everyone. So if I can invite Bill and Carl and Kenneth to join us on these comfy chairs, as it were, on the chairs, and indeed, as that there are speakers hidden in the green room, as it were, in the panel, who are going to appear later this afternoon. And any of those who we've not yet heard from, who would like to join in discussing what we've heard so far, please do feel free to do so. And anyone who's got some slides can screen share in terms of any of the answers to questions. There are a few particular detailed questions. I'm going to leave speakers to handle them. What did you mean by this? What are we measuring there? Perhaps online, and you can type answers. But I wondered if we might discuss just for a little bit. Those of you who contributed so far, it seems to me that we have three worlds that there's only one world, three ways of looking at the one world of brains and perceptive modules in brains. We've got we have the neural net world of Bill and Kenneth, and dimensionalities, and massively large numbers of neurons, and correlations that seem to be sufficient to understanding this as a machine, but of course that's a machine, or that's a system for a purpose, and its purpose is to make sense of the world, which brings us to the second perception, which is perceptual perspective, which is Carl's, of if I call it Bayesian influence, with knobs on. I mean, it's an algorithm that looks like a free energy that I'm sure Bill would tell us it's not a free, perhaps it really is a free energy. I don't know. But this sort of learning algorithm at a higher level. And then we have an example of a specific perceptual task that Daniel's told us about, and a sort of Schrödinger equation, perceptual psychophysicists, Schrödinger, that is, not the quantum mechanical Schrödinger equation level of description. Now I wondered, do we have how big is the job on our hands to connect those three worlds? So that's my discuss for 2 minutes question. Anyone want to have a go?
I mean, I could take a quick try.
We'll have a go, yeah.
So, I mean, this is a somewhat personal answer. So for me, some fraction of my time is spent on things that are more the flavor of what you heard from Carl and Daniel, of trying to use optimization principles to understand why functional behaviors are the way they are. And I think that if you want to do that, you really do want to drive, you want to spend a certain amount of time at the abstract level, but you do want to drive down to very particular computations that are important for, processing, signals that animals actually care about in specific examples. And then there's this other part where, which is what I talked about today, of saying, well, let's look at this very large network of neurons, or, I've done related things in other systems, and let's let the data drive us, right? And for me, I try to keep those two parts of obviously these have to be related to each other, right? The collective behavior of the network is there to implement certain functions. And so these have to be connected. But at least for me, I've kept them separate because I don't want my thinking about what the data is driving us toward to be influenced by my ideas about what are the principles that select particular behaviors. I want to keep those, so for example, being at a fixed point as a normalization group is equivalent in some sense to being at a critical point. There is an enormous literature about why it's a very good idea for the brain to be at a critical point. There is much more literature about why it's a good idea than really testing the idea itself. And so there's a great danger that, you find things, and since you know why it should be true, you believe in it more. And so that's why, at least for me, that's why those two worlds are separate. Obviously they better be related, but at the moment I find it useful to keep them apart.
Sure. Ken, you're nodding, but it looks as if you have nod and want to say something.
No, I just agree completely with that. I think it's a real danger in this field that we have. A long time ago, I started but did not finish a PhD in string theory, and that's an example, I would say, hopefully not offending anyone, of a field that was so beautiful it just had to be true. The equations are so beautiful they have to be true, except it now seems like they probably aren't. And that's a real danger we have in neuroscience as well. We can have these theories that are so beautiful they have to be true, but that doesn't mean they actually are. And I think Bill made a really good point there, that's why it's really important to let the data speak for themselves.
We also have the huge advantage that although the experiments are phenomenally difficult and to get good data is very hard, it is not, as in the case of string theory, completely impossible.
Yeah, and there's so much data sitting around just on hard drives not being analyzed right now.
I mean, another simple way to say it is you can theorize top down and you can theorize bottom up, and they're both productive, but you should be sure you know which one you're doing. So
absolutely. So I mean, in terms of critical points, it occurred to me that critical points are, to me, as a statistical physicist, also connected with scaling. Kenneth shows a beautiful example of what looked like 1/f noise to me. I mean, 1/f plus something. is that exponent connected in any way? I'm not thinking fast enough to work out my answer, my question to Bill's work's discovery of of scale invariance.
Yeah, I've never, I mean, I've been giving this talk for a few years now, and I always get the question how does this relate to critical phenomena? And I always said, I don't know, but I thought Bill's talk was really, really interesting from that perspective, because it actually may be connected, and I'd love to learn more about that. So I'm going to read PCA meets RG. I think that's probably the place to start.
Yeah, I mean, in principle, one could do a PCA analysis of Bill's models, right? You've got.
So in fact, we've done the course. So another way of doing coarse graining is rotate into the space of PCAs, average over the components that contribute the small variance, and then come back. Right? That's also coarse graining. That actually, so for the physicists in the audience, right? You can do the normalization group in real space, or you can do it in momentum space. Right? You can do blocking, or you can do momentum shells. So rotating to PCA, averaging over the small variance components, and rotating back is equivalent to momentum shell normalization. And you see things show up, things that are supposed to be the same come out the same. Obviously, certain probability distributions look different because of what you've done. But you see the same notion of approaching a fixed point and so on.
So in terms of, I mean, all these good reasons why it would be good for brains to be at a critical point, it worries me that we have too many good reasons for this to be true, and too many possible versions of what critical points there might be. What, in terms of hard getting at the data, doing like Daniel's experiments, what are the experimental programs that will tease this out and ask, well, what is the good it may be just the hippocampus. Obviously, does the visual cortex have even the same scaling behavior as the hippocampus does? And perhaps it does, perhaps it doesn't. But for those parts of brains or subsystems that have shown scaling, can we pin down the good reason that evolution has pushed them to that place, and how would we do that?
So just trying to connect in real time what Bill said to the way I've been thinking about this, Paolo, to the way Bill thinks about it, which I think is slightly different. The way I've always thought about it is that it's kind of what you're going to get when the neurons that you happen to be recording are drawn from a probability distribution. There's a vast number of neurons, and you just recorded 10,000 of them, and actually the brain only contains a few million out of this potentially infinite number of them. I'm just trying to connect this to the idea of scaling. Perhaps one way to think about it is, what would be a mechanism that could allow the brain to look like that? So you've got to have a network mechanism that makes it appear that the neurons are sampled from an infinite population, and some sort of system where you could kill half the neurons and it wouldn't really affect things very much, other than reducing the number of neurons you have, maybe is somehow, in a hand wavy sort of way, connected to this idea of the renormalization group, that you'd, in a sense, average them out. So, I mean, just potentially, that's one potential experiment would involve taking a neural circuit and removing a large number of the elements from it and seeing what happens. And that would do a sort of a scaling, in a sense. And if it's robust to that, that's maybe some evidence for scaling.
Right. So it would be an argument for robustness or resilience.
Yeah.
There's a question from Tony Morland. So some of our other panelists, of course, aren't able to ask questions in the Q&A. So they come through our private little chat we got on this side for the conference. And Tony asks, Kenneth and Bill, are there features, parameters in the models that could capture pathology or aging of the brain? And if so, is there potential to detect pathology early?
Well, if you're going to do that in a human, it needs to be done from the outside of the head. So you're pretty much restricted to EEG or fMRI. I'm not the best person to answer this. I mean, I think there are signs of Alzheimer's and EEG, the slowing of the rhythms, but I think if they were really that good, they'd be used by now. I mean, maybe someone else knows more about this.
I wonder whether Tony's thinking about sort of the de-differentiation you get in aging, which certainly does, I think, if you look at coarse graining as a way of parceling the brain at different spatial scales and a measure of the functional specialization that is certainly lost in aging and dementia and certain neurodegenerative conditions. So one would anticipate a different kind of scaling exponent using a sort of coarse graining approach, in neurodegeneration and aging.
Interesting. I mean, other ways of getting at differences in these sorts of models from the outside, and sort of psychophysics, of course, I mean, you think of Daniel. I was going to ask Daniel whether, I mean, the eyes sometimes give you, the eye, because of its cones and rods, actually give us ways into different networks without having to inside. So I wondered whether what happened in your results if you selectively fired up in a binocular way, you've done short, medium, or long wave cones, for example.
Well, that's a really great question, and it just so happens that I've put in a grant proposal on exactly this, which hopefully the BBSSC will decide to fund. I think their panel meeting is on Monday. But yeah, I mean, that's a really interesting point, that actually we have in the brain multiple pathways that combine signals across the eyes. So it doesn't just happen in primary visual cortex, which is of course what we teach undergraduates. Actually, there's also a load of subcortical nuclei that combine, for example, light signals to determine how big your pupils need to be, how dilated your pupils need to be. And in fact, even more excitingly, there are some subcortical pathways which determine melatonin production, which influences how sleepy you are. And that's also driven by light signals from the eyes. We don't really know very much at all about how signals are combined binocularly in those other pathways, because most people so far have focused on the cortex. So yeah, you're right. There are many sorts of, the brain does all sorts of different interesting things. But yes, good question. Maybe if this grant gets funded, I'll have a better answer in 3 years.
If Hannah, who's talking in the second session, working with her a little bit, has taught me that's a question I wouldn't have known to ask it before. I've been working with her for a bit. Thanks, Hannah. And the answer to Tony's question, says Steve Smythe, is come to the neurodegeneration workshop on February the 10th. Our speakers will touch on that. Steve has been also directly messaging me with just a prompt. I think we've just got time to finish this, which takes us back to our first question, or the Bill's answer to the first question, which is to be careful whether we're working in top-down mode or bottom-up mode, and to keep be clear about which way we're working, but also to say at some point, if you want a complete picture of the brain, these have to join up. So and I think you said that, Bill. So the question is, where would they join up, and with what data, in what experiments, and in what sort of models would top down and bottom up join up? Carl, have a go. Yes, anyone?
I just wanted to speak against the this sort of dichotomy between the top down and the bottom up, because I think there's lots of points of consilience between the sort of two pairs of talks. So, for example, the omegas in the visual Schrodinger equation would be known as Kalman gains, as was noted at the end of the presentation, and they are exactly the gradient flows that you get from this top-down variational free energy minimization, often cast in terms of precision. So I'm just mentioning this because there's actually a vast literature out there on pathologies of encoding precision in a lot of neurological and psychiatric populations, and in particular as an explanation for visual hallucinosis in certain forms of neurodegenerative disease. So I see there is actually a lovely connection between sort of, the dynamics of a brain cast as a gradient flow on the right objective function, which is where I was coming from, in particular the ensemble dynamics, and the way the brain encodes certain sufficient statistics, like the second order correlations that Bill was talking about. From my point of view, you were asking, what's the reason for this? Why is criticality useful and has a high adaptive fitness? So the answer, if you're subscribing to the free energy principle, is that it minimizes or optimizes free energy. What does that mean? Well, in virtue of the fact you've got an entropy term there, you're going to have basically a low curvature of your free energy landscape, which leads naturally to criticality. So the answer to your question is that your adaptive fitness is low free energy. What does that entail? Well, by construction, it entails critical slowing in virtue of the Occam's principle part of the free energy principle. So then the game is to say, well, how is that actually instantiated in neuronal ensembles? And again, I think that it's actually a very easy job. In fact, the other half of my job in brain imaging is exactly to do this. It's to take empirical measurements of neuronal dynamics and understand them as the gradient flows that subtend non-equilibrium steady states. So I'm much more sort of optimistic that these will meet, and are meeting all the time in certain fields.
Great. Well, thank you, Carl, very much. At that point, I think that's a very good point to round off this fascinating first half day. Thank you so much. And so that Anthony Moreland has a good 3:30 start for the session on imaging and techniques, and so that people have a 4 minute now chance to have a cup of tea and a quick break. So again, let's all thank those of us who can visibly do so. Bill, Kenneth, Carl, Daniel, for kicking us off so wonderfully, for leaving us some great questions, and we'll reconvene 3:30 under the eye of my colleague Anthony Moreland. See you then.
Thank you. Bye-bye.
Bye.
Hello, everybody. Welcome back. I'd like to welcome you to the neuroimaging measurement and other techniques session. Seems we might have had some other plans on our minds when we thought of that title, other techniques. Yes. Okay, I'd like to thank all the organisers. This is a fantastic meeting so far, and I hope it continues to be so, and also thank the speakers for this session. We've got some diverse topics, and I think it's fair to say that no single person could be an expert in all of them, and that's just exactly what the sort of secret recipe for this, or not so secret recipe for this workshop really is to get our minds whirring and be exposed to things that we wouldn't normally be exposed to if we were to go to our regular conferences. Just as before, if you'd like to ask questions as you go along through the Q&A channel, that's absolutely fine. We might be able to to answer some of those quickly after the speakers finish, but if you've got outstanding questions and more perhaps deep and meaningful questions, we can save those for the panel discussion. That's it from me. I'd like to invite now Amanda Ellison from the Psychology Department at Durham to open up, and she's going to be talking about this. She's going to say, if the physics of life is a powerful beat, what does the rhythm of the brain tell us? Over to you, Amanda. Thanks very much.
Thanks so much, Tony. Thanks for inviting me to speak at this meeting. It's allowed me to unapologetically indulge a latent passion of mine for physics. It's been utterly fascinating so far, but at the danger of introducing life sciences, I'd like to speak today about human brain rhythms and their importance for understanding our brain, body, and behavior axis. It leads on quite nicely from Daniel's talk, really, but perhaps at a different scale. Let me just press some buttons here. But first, here's some background. Now, the way we look at the brain and how it becomes active and how it does that has evolved over the years. Based on theories going back to Gall at the turn of the 19th century, we were already thinking about the brain containing multiple and distinct faculties. This is the earliest form of functional localization. And unfortunately, this led to the idea that we could quantify the development of these known faculties based on the lumps and bumps of your skull. And this is known as phrenology, but it was a huge misstep in brain science. Happily, there were others around the same time who were forming more solid conclusions from the observations that they were making. This is Brodmann, who is interested in cytoarchitecture, and by grouping cells that look the same, he theorized that these groupings must do the same thing. In much the same era, Karl Kleist was investigating World War I head wounds and the functional loss that resulted, which we now call neuropsychology. So taken together, this further worked towards the validity of functional localization. Now, of course, when people have brain damage, the area affected is rarely discrete, and there's the danger of misattributing functions or missing nuances with respect to what areas do. Modern neuroscientific techniques such as fMRI can be more exact. With this technique, we have to be mindful, however, that just because a brain region is active in the scanner, it doesn't mean that function originates or is localized there. Less correlative is our use of transcranial magnetic stimulation, which allows us to briefly and reversibly disrupt the activity of neurons below the magnetic coil. Working via the premise of electromagnetic induction, which of course you're all familiar with, neurons under the coil are activated. And if you do this over motor regions, for example, you will get part of the body to move. And if you stimulate a more enigmatic region of the brain, you can probe its involvement in a task. Because that area is switched on in response to the magnetic pulse, it can't properly process the task your participant may be doing at the same time. Its spatial specificity is better than electroencephalography, and its temporal specificity is better than fMRI. Crucially, if you move your coil even ½ centimeter away, it can stop having an effect. And that's another big tick in the functional localization column. But of course, we know from old fluorotracer experiments done on chimpanzees in the early 1950s by Bailey and Van Bonnen, and the more modern diffusion tensor imaging techniques, that neurons in the brain are very connected, and this happens often over relatively long distances. So what does this mean for function? Well, it means that even though different regions have different specialities, in order for us to behave in the seamless way to which we have become accustomed, these regions must talk to each other. This brings us from a functional localization concept to one that's better represented by functional connectivity. This is a graphic of the connections involved in only the visual system that denotes a hierarchy of importance, of integration, but also of feedback from high-level processing to the very lowest levels in the brain. And this is the mechanism of attention, for example. So once we start to look at brain function in this way, we need to start to think about how these brain regions work together, if they do at all. And we've talked about what fMRI can tell us about that, but it's very correlative. By sending magnetic pulses into multiple discrete areas of the brain at the same time, though, we can begin to see that for certain functions, there may be two regions that are important, and they must communicate with each other in order to bring about efficient function. So in this case here, we have an interaction between V5, which is a visual area important in detecting movement in the visual scene, and the parietal cortex, which is important in the deployment of attention. Now, essentially, V5 is sufficient for a visual search task that requires attention to motion all by itself. PPC isn't involved in this. By PPC, I mean here posterior parietal cortex. It's not needed at all. But if we disrupt V5 using transcranial magnetic stimulation, then parietal cortex is required to help out, as denoted by the large additive effect in the reaction time taken to complete each trial, when both are knocked out of the processing loop. So we see recruitment happening when the brain isn't working optimally. Here's another example. It's a little bit different. In this case, an area involved in shape identification, which is lateral occipital region, and an area of the parietal lobe important in spatial discrimination. Each are pretty equally involved in the processing of a task, where one must decide which of these boxes is closest to the central box. So each can do it on their own according to their own functional specialities. But cooperation is revealed if we stimulate both together with an additive effect on reaction time, slowing processing down even further. Now, contingency can be inferred by the relative peak of activation of each of these regions, with LO and V5 peaking earlier than parietal regions, making it reasonable to assume that our parietal region is contingent upon signal coming from these areas. But more concrete proof comes from when we play with the activity of a region and then scan the brain to see where else is affected. We use transcranial direct current stimulation. This is a technique that has its detractors, but using it in this kind of multi-technical study can give you insights that have been impossible in the awake functioning human before. Here we see that if you put a cathode, which decreases the activity of an area by making neurons less likely to generate action potentials, over a parietal area, whilst doing a task it's usually involved in, you get concomitant decreases in activity in frontal regions. So using TDCS particularly, you have to be careful what inferences you can make about the specificity of effect of stimulation to just one area. Given its relatively longer timescale to TMS, remember event-related TMS runs in the milliseconds, TDCS runs for tens of minutes. Results can easily be driven by distal effects on other regions of the brain. However, this makes a really important point for neurorehabilitation, because compensatory mechanisms that are built upon recruitment of spared regions may be destined to fail if signal is compromised from the primary region of damage. Okay, so far we have established that areas of the brain have specializations, but that they fruitfully work together to bring about seamless behavior that we rely upon for our daily lives. Well, how do they do this? Well, rhythm is the dancer. It's the soul companion. You can feel it everywhere. I'm showing my age now, of course, but that's what the song tells us. As a neuroscientist, I don't disagree. I've already mentioned that the direction of this rhythm can be determined by the peak involvement of different regions of the same task. This is our LO and PPC peaks, where LO is relatively quieter than PPC early doors, but both pick up later in the processing loop, denoting that shared involvement that we talked about. But the critical issue is the rhythm at which neurons communicate. We use electroencephalography for this, and by amplifying the electrical activity that's happening in the brain, we can see discernible rhythms emerging. This was invented by Hans Berger in 1924 with a willing guinea pig in the form of his son Fritz in his garage. He was incredibly sensitive about his work, giving lectures on telepathy in his day job, but I digress. Thanks to Hans Berger and others, we now know quite a lot about brain rhythms involved when we are asleep, and these are driven by the thalamus. Now, if you get a chance, please do look up the Durham University Institute of Advanced Studies Threshold Worlds theme, which looks at dreaming, which of course can happen in any phase of sleep and not just rapid eye movement sleep, and issues related to permeability, liminality, narrativity, immersivity, and reportability in this unconscious state. So here we see clearly defined phases and rhythms in sleep, each with separable functions. Two to point out here are sleep spindles, a rhythm set by the thalamus to synchronize activity across the brain, and K complexes that integrate information from inside and outside of the body. So this is the reason why a loud bang can wake us up from our slumber. Both are thought to be important in memory consolidation and emotional regulation. The point, and we'll come back to it, is that the thalamus is very important in setting the rhythm and pattern of firing rate across the brain during sleep. But what about when we're awake? We know that being able to entrain brain rhythms can positively affect our behavior. Buddhist monks and people who meditate have been doing this for centuries, integrating gamma wave activity across diffuse regions of the brain, leading to greater emotional control and focus. Synchronized gamma rhythms at bursts of 30 to 80 hertz in areas across the cortex associated with binding various neural components into a single perceptual construct. We might call it a shortcut to clarity. But again, this is a thalamic function. And it turns out that there are a number of thalamocortical loops relating to a number of different functions encompassing sensation, vision, audition, emotion, movement, all of these. Each can have different oscillatory rhythms. This is something Rodolfo Linus and his team recognized in the late 1980s and progressed through the 90s, showing that damage to or abnormal activity of any of these loops can have functionally significant effects as determined by magnetoencephalography. Particularly, he recognized that the firing rate was faithful between an area of cortex and the thalamic loop that served it. For example, if the thalamus is sending a slow pattern of firing to the motor cortex, this is replicated in the 3 to 6 twitch per second frequency seen in Parkinson's patients. It's worth pointing out that thalamocortical dysrhythmia, as this is known, is a symptom in disorders varying from Parkinson's disease, pain, migraine, through to tinnitus. However, it does seem to underlie the symptomology. So if these can be addressed, symptoms are ameliorated, whatever the causality is. This immediately created a step change in treatment, leading to neurosurgeons of the day, such as Mark Chenminot, ablating the areas sending the sleep-like waveforms on the proviso that you're better off having no signals than bad signals. And there was some success with this, particularly in dystonia. However, surgery was too hit and miss. With the advent of technologies allowing surgeons to detect the abnormal signal and insert a pacemaker to entrain normal rhythmic firing rates, deep brain stimulation has become a viable treatment candidate for many disabling disorders. However, entraining brain rhythms can have advantages in the neurotypical human too. Such entrainment happens naturally via environmental rhythms, such as the circadian rhythm, as we've talked about, with light coming in through the eye, or even breathing patterns and the receipt of sensory inputs such as speech, as this graphic shows from a lovely review by Grigor Thut and colleagues. What I'm most interested in is how I can artificially entrain brain rhythms using transcranial stimulation of the cortex, which may be magnetic or it may be electrical. For instance, rhythmic forms of TMS at alpha frequencies over parietal areas cause visual perception to fluctuate in an alpha cycle. That is, to entrain at an alpha frequency, thus improving function. Similarly, simultaneously transcranial alternating current EEG and MEG studies in humans have shown that this can entrain rhythms affecting sensory, motor, and cognitive function. For example, transcranial alternating current stimulation of auditory areas benefits perception of serial auditory events if it is phase locked with the input. And that's in line with the idea that the frequency of brain oscillations causally determines the rate of sensory sampling. However, effect sizes are small, and there are other problems in these studies which have led to the premature proclamation of human enhancement. But it is in rehabilitation following brain injury that we'll see the most benefit here, both in terms of hastening functional recovery or compensation following damage to perceptual, motor, and possibly cognitive domains. There's been some success in Alzheimer's, for example. As mentioned previously, oscillatory firing rates are disrupted to a hitherto largely unquantified degree, not only feeding into the primary functional loss, but also undoubtedly underlying the social effects that we see in such patients, such as altered sensitivity to loneliness or happiness. We need a personalized approach here, as one size certainly does not fit all. So for my last slide, I would like to pose a bridge with another concept, and that of quantum physics and how that is conceptualized. We know that our brain's activity and its oscillatory patterns may be generated by our subcortical structures, like our thalamus. In real life, these patterns are entrained by our environment and our sensory world. In quantum theory, and I hold my hand up here as an eager to learn non-expert here, a unifying description is that our reality is shaped by the question we ask of it. By a minor extension, our reality is what we are conscious of, but until we are conscious of it, all possibilities are possible. Now, consciousness is often described as a filter of our internal processing, and we have discussed brain rhythms in relation to that. We need to discount what is irrelevant, such as self-produced speech, in which case entrainment can decouple the receiver from the rhythmic input stream. If the entrainment breaks down, the inner voice becomes relevant and is interpreted as being an external voice. This issue at hand is how it is that we interpret the coordinated activity in the brain. In dreaming sleep, the alpha activity is interpreted as a conscious experience, even though we are in effect paralyzed, certainly at least during rapid eye movement sleep, when the activity of the visual areas is decoupled from the frontal lobe, which means what we see doesn't often make sense. This is analogous to how a magnetic pulse delivered when you're asleep does not have the same diffuse effects as it would when we are awake. The induced activity remains localized, but what reaches consciousness, or at least our memory of a conscious percept, determines what we perceive or do, or to think what we think. We know that we can act without consciousness, of course. Priming and blindsight tell us that. But it's likely our preconscious may be a background processing state with an oscillatory rhythm, whereby all possibilities are possible. What we have not defined yet is at what point this activity becomes cohesive, such that it is perceived as a singular consciousness. Given our discussion of rhythmicity across the brain, let me leave you with this. Is it not reasonable to argue that those who experience hallucinations or have various personality disorders, they may have a broken filter, whereby usually preconscious and thereby ignored processing comes to the fore and creates a new and alternate reality for the individual? For example, I've mentioned the lack of entrainment with inner speech, and that may lead to the experience of an inner voice as an outer voice. I would propose the thalamus, and particularly spindle type waveforms that may hold the key in a physiological sense to this psychological construct. But despite good theoretical company with Penrose and others, this is not about asking if the brain uses quantum mechanics in terms of superposition or microtubules or Posner molecules. What I want to do is dance between the raindrops and define the forest from the trees first, embracing Occam's razor, in which the question remains, can we use quantum theory approach to inform our gross thoughts on consciousness? So that's my question. I'll leave it there, but I'd like to thank all of these people who have suffered upon me talking to them about this over the past too many years to mention. Thank you very much.
Thanks so much, Amanda. That was a real tour de force, and I really enjoyed it. There are no questions in the Q&A session. I'm sure that some will emerge later. So you stuck almost perfectly to time. There's one thing that we've got, just a fraction before we start on the next talk. Now, I was really interested in, and this is probably speaking to my ignorance rather than insight, is that we have these highly correlated neural activations in these rhythms. Whereas in the previous session, we were talking a lot about the efficiency of orthogonality of representation. And I mean, of course, we're measuring slightly different things. The sort of the whole orchestra is what we're looking at in the EEG, whereas what we were looking at before was a different measurement in looking at a single neuron spiking activity and so forth. But I've always struggled to reconcile those two things, sort of orthogonality and then the highly correlated electrical potentials. And I just wondered if you had any insights on that. This is where it's just so interesting in that it's the interdisciplinarity of these two fields. I looked at, in fact, I confess I took a screenshot of one of Carl's slides, and it's very much, we talk about the same thing, but we use very, very different languages. And we speak of it at very, very different scales. And I think that's the value of a meeting such as this, that we can actually put those two things together and actually start to say, okay, are we talking about the same thing? Yes. Are we talking about different scales? Yes. But how do we bring them together? So I'm not going to answer that question very deftly, I would say, but I hope that we actually do get to that over the next 2 days. Okay, I'm glad you answered in the way you did, because you didn't embarrass me, at least. So thanks very much. Okay, We'll move on now to one of our locals, or local to me at least, at the University of York, David Halliday. He's going to talk about neuromorphic computing systems, spiking neural networks, astrocyte neuron networks. Over to you. Thanks very much, David.
Okay, thanks for the introduction, Tony. And I'd also like to thank the organisers for putting this meeting together with such a wide range of topics to talk about. So I'd like to talk about neuromorphic computing systems, focusing on spiking neural networks, and in particular, some of the recent developments looking at regulation of neuronal activity by networks of astrocytes. So we'll say a little bit about neuromorphic computing with spiking neural networks. We'll introduce the idea of astrocyte regulation of neuronal activity, starting with the tripartite synapse model, where we have the three components involved. We'll see how we can implement that system in hardware and how it can be used to give us a fault-tolerant mechanism for synaptic transmission. Then we'll report on some recent work extending that, looking at how we can have self-tuning and self-repair in spiking neural networks using the quadpartite synapse model, where we extend the system to look at what happens when we include an inhibitory interneuron in the four-way interaction at the synapse. And finally, we'll show how we can build resilient hardware systems using robotic demonstrators. Resilience in this case, we mean has embedded fault tolerance and is able to correctly map inputs to outputs from sensors to actuators. So with respect to neuromorphic computing, if we back up to the first slide just for a moment, then the figure on the right there, this circuit board, is the IBM True North neuromorphic computing system. This was launched about 5 or 6 years ago and is one of the now several commercial available neuromorphic computing systems. So what you're looking at there, each of those yellow squares is a single chip on the board. Each chip consists of 4,096 neurosynaptic cores. And the neurosynaptic core contains 256 spiking neurons. So this is based loosely on the idea of a cortical column, where we've got sort of the order of 200 to 300 neurons in a single cortical column. As part of the design, they have this fully interconnectable connection. So each of the 256 neurons can flexibly connect to any of the others in each neurosynaptic core. So on a single chip with the 4,096 cores, you've got 1 million spiking neurons and 256 million connections. There are 16 chips on the boards. Of course, that gives us 16 million neurons and about 4 billion connections. So you can see now that these systems are beginning to operate at scale. Okay, so moving on then to think about computing with spiking neural networks. So this was a paper published in 1997, quite an influential paper now, and it was one of the first to really introduce the idea of thinking about spiking neuron models, not just as a way of modeling the behavior of the nervous system, but as computational entities in their own right, drawing on the success of conventional, artificial, or what we might call analog neural networks. And for that reason, they're termed the third generation of neural networks. The first generation, of course, being McCulloch and Pitts binary units, second generation sigmoidal or continuous output analog units, and the third generation being the spiking models that we're interested in. And of course, computation through spikes is fundamental to understanding the brain. And this figure, although it's quite old now, this figure I think is quite a useful reference point. As we look at it, we can see it's a raster plot with time along the bottom and then individual neurons identified up the axis. And each vertical line represents the time of firing of an individual neuron. So what can we take from this figure regarding principles of neuromorphic computing? Well, we can see that there are two key ideas here. First is that the spikes are quite sparse. So a lot of the time the neurons aren't really generating spiking activity. Of course, intracellularly, there may be a lot going on, but extracellularly, they're quiet for much of the time. The second component we can take is the stochastic nature of the firing. Firing patterns to first approximation appear fairly random. And we've heard a lot in the previous talks about the importance of statistical models for representing, analyzing, characterizing neuronal activity. So if we're going to work with spiking neural networks in our neuromorphic computing systems, then we have to be able to think about how information is represented through spike timings, and in particular, how that relates to the communication between neurons. So in spiking neuron models, the communication is through synaptic connections. So this diagram shows one of the key ideas that's coming forward now in terms of the astrocyte regulation of neuronal activity. What we're looking at in this diagram, we can see vertically here, we've got the presynaptic neuron and the postsynaptic neuron with the synaptic cleft in the middle. The circles represent the various molecular signaling agents that are present in the system. The squares represent receptor sites for binding of these, and the arrows represent the pathways involved. So we have the classic excitatory glutamate-mediated synaptic connection here, going from pre to postsynaptic. And in most spiking neural networks, that's treated as being the synaptic connection. In the tripartite synapse model, it's treated stochastically. So we associate a PR, as it says here, a probability of release with each individual presynaptic spike. We have these two feedback pathways. We have this direct pathway here, where this is mediated by 2AG, this molecular signaling agent. And this causes a depression of the excitatory activity. So it's the tendency to depress. It's a kind of negative feedback regulating the probability of release. Mediated by the astrocyte, we have this indirect pathway. And we can see it's mediated by IP3, and then there's calcium activity in the astrocyte, calcium oscillations with the threshold and so on, quite complex dynamics, but the net effect is that we have a release of glutamate. So again, that gives us an excitatory pathway. We have the balance between the inhibitory direct connection and the excitatory indirect connection. So these two act to regulate the probability of release at an individual synapse. So I haven't given the full name for these agents. You can find a more detailed description in the Wade publication there, giving a bit more details about the biophysics of what's going on there. But from our perspective, we're interested in what happens when there's a fault at this synapse. So we have the situation where the indirect component is preserved through the astrocytes, although the direct connection fails, and this direct feedback pathway no longer is active, then the indirect pathway is still active, acting to restore the synaptic connection, maintain the probability of release. So we can see that in this simple example here. So what we're looking at, the green represents two individual spiking neurons. We model these with simple leaky integrated fire dynamics. We have a single astrocyte. We've got the direct pathway, this DSE, direct suppression of excitation, and the ESP, the endocannabinoid synaptic potentiation through the astrocyte. And those two act to balance out the system. So what happens when we inject a failure? So we're interested, of course, in the design of electronic systems. So we're looking at implementation on FPGA, field programmable gate array systems, essentially programmable electronic systems. So if we give a synthetic lesion to 80% of the inputs to neuron 1, so each neuron, as it says, has got 10 synaptic inputs, then we can see the time scale here. About halfway through, at 1 millisecond, we inject the fault, and you can see there's a transient dip in the firing rate of the neuron, but then activation through the indirect signaling acts to recover the information. We can also see in this diagram the decrease in the firing rate as the initial regulation of the inhibitory and excitatory components work together. I should also say in the time scale in this, we're running this in hardware in real time at 100 megahertz. The time bin we use in the integration for the LIFT model is equivalent to 1 millisecond. So we've got about 100,000 times speed up in terms of running this in accelerated timescale, which is why the timescales here appear very short and the numbers of spikes here appear very large. But essentially, what we've got now is a system that's giving us fault-tolerant, self-regulating pathways in our spiking neural networks. So let's move on and have a look at some more recent work in terms of how we can really build on the new knowledge that's coming now from neuroscientific studies on astrocyte regulation of neuronal activity. And the schematic here is meant to represent this new enhanced synaptic connection model. So we've got this circle containing the astrocyte, and of course the astrocyte connects to a number of neurons, and we've got this inhibitory interneuron sitting there as well, contributing to the regulatory process. So we extend the tripartite synapse to build up this additional component, this GABA interneuron. So we've now got an excitatory effect. I beg your pardon, an inhibitory component to the synaptic connection. And we look to see how we can combine traditional STDP spike time dependent plasticity, where it's the timing of the pre and postsynaptic spikes that determines whether the weight is potentiated or inhibited. We combine that with a firing rate representation based on the Bienenstock-Cooper-Monro rule for selectivity, which models the effect of pre and postsynaptic firing rate. So let's have a look then at how we can build up this new model. So quite a busy figure here, but there's two sections, top and bottom. At the top, we can see we've got the presynaptic axon on the left there and the postsynaptic on the right. We've got the astrocyte as before, and we also have now this GABA inhibitory neuron. But low firing rates, then the effect of the inhibitory neuron dominates. It's inhibitory, releasing GABA, so that tends to suppress the effect on the synapse. So the PR, this probability of release, is quite low. So as we build up the shape of this graph here, which is the presynaptic firing rate, F3, against the postsynaptic probability of release, rather, PR, we can see that it's quite low. At the lower panel now, what happens when we see an increased firing rate in the presynaptic neuron? Well, this is sufficient now to activate this calcium threshold inside the astrocyte, leading to the release of the glutamate, overriding the inhibitory effect of the GABA interneuron. So we can see now that the PR is increasing. So the probability of release is increasing as the firing rate moves towards the center of this tuning curve. And of course, at higher firing rates, what happens? Well, the calcium diminishes, the calcium transient stops. So the inhibitory effect of the GABA tends to dominate again. So we get this decrease. So we have this classic Gaussian shape. So we model this using a Gaussian effect. So it's tuned to this center frequency, F pre. So this is the presynaptic firing rate and the probability of release. We can use the postsynaptic firing rate to determine whether we want to potentiate or depress the synapse. So this is the classic curve we see in the BCM rule. Here it's tuned to a center frequency of 25 spikes per second. And the idea is once we reach this tuning rate, we get a transition, this A0, which is the height of the potentiation window used in the STDP rule. This swaps from positive for potentiation to negative for depression. So if the output firing rate is too high, we get depression, so it tends to bring the weight down. If the output firing rate is too low, we get potentiation, that tends to increase the weight. So this component is tuning the response of the neuron to a target output firing rate. And of course, sitting between these presynaptic input and output firing rates, we've got the STDP rule, where it's the timing of the spikes which determines how rapidly the weight is potentiated, going back to Donald Hebb's idea of building these neural assemblies. So with this rule, we can then construct our networks where we've got simple input-output mappings, and these verification networks, we use binary representation. So we've got two states mapped by a low and a high firing rate for the neurons. And these tuning curves represent these Gaussian windows, giving us a regulation of the synapse and causing the correct mapping determined by the target center frequencies of each of these from input to output. So we can select a particular action. So for example, for a robot with a sensor, a proximity sensor, we might want to signal avoidance of a particular obstacle. And with a target sensor, for example, a video camera, then we might say we want to approach a target object. And of course, sitting on top of this is the fine-grained fault tolerance that we see in the tripartite synapse. So that's still embedded in this quadpartite synapse learning rule. So let's have a look at some results now from this. So this is some initial results, again, looking at hardware implementation. So we've got a simple 4-neuron network, neurons N1, N2, and N3, providing input, mapped onto single neuron N4. And we've got the low and high firing rates given here as numbers of spikes in a particular time window. And the system is stabilized. We've got an output signaling that we've got the correct pattern of inputs. And in each of the connections between N1, N2, and N3, we've got 8 synapses. So we synthetically lesion these systematically, one at a time, shown schematically at the top for neuron N1, neuron N2, and neuron N3. And you can see each time there is a lesion, we get a small dip in the firing rate, but it recovers quite rapidly. This is the self-regulating effect. And notice that it goes back to the target rate of 54 spikes per time window. It takes a little longer, but at this point, we've only got 3 of the 24 synaptic connections remaining. And in the lower panel, you can see the weights being selectively increased for neuron N1, neuron N2, and neuron N3, which is the last neuron, so the weights again are comparatively bigger than neuron N2 and N1 as the synthetic lesion takes place. So this is demonstrating the system working in hardware to maintain the output firing rate in the presence of injected faults. So this is an important design property for electronic systems, this idea of having distributed fine-grained fault tolerance, but also part of a neuromorphic computing system. Remember, the signaling between the components here is the signaling that we see in synaptic pathways. So in a final application, which I'll just summarize very briefly, we're verifying it using a simple robotic demonstrator, these mobile robots here. So we've got a leader here with a QR code on the back and the follower with its camera trying to follow this. So this setup is verifying the target tracking system. So in the target tracking system, on the right, we can see 2 networks, target tracking and obstacle avoidance. So we are verifying that the tracking system is working. We've got proximity sensors for forward, left, and right, and again, target detection for forward, left, and right. And we prioritize the target tracking. So this system now allows us to construct mobile robots using neuromorphic neurocontrol systems, which are robust to failures in the synaptic pathways connecting the neurons in the network. So we're working towards the concepts that might be important for the next generation of neuromorphic computing systems. So I'll just finish up by thanking the colleagues and particularly the RAs who worked in the project. So Andrew Johnson, Alan Miller, Devon Scissor, and Maciej Napora at York, Jingsu Liu and Siobhan Karim at the University of Ulster, and also acknowledge the funding from the EPSRC. Okay, so that's me finished, so thank you.
Thanks for that. I'm just looking at the Q&A and I haven't got anything upcoming as yet, but as I said, panel discussion may be a place to have a more extensive discussion. Just in the interest of time then, I'll thank you again and we'll move on to Hannah Smithson from Oxford.
Okay, thank you.
Thanks. So welcome, Hannah. And Hannah's going to be giving a talk entitled Looking into the Brain, High Resolution Imaging of the Living Human Eye. Over to you, Hannah. Thanks.
Great. Thanks very much for the introduction, and thanks for the invitation to speak. So yes, I'm going to be talking about high resolution imaging of the living human eye. I just also wanted to introduce my group. So just to say right at the beginning, all of these people have contributed to the work I'm going to talk about. I'm obviously talking in a workshop on brains. And so I wanted to start perhaps with a little bit of a discussion about why I'm going to talk to you about the retina. Obviously, the retina is a piece of neural tissue, but it's a relatively accessible piece of neural tissue. And so it's particularly suitable for non-invasive optical imaging. And we know a lot about its anatomy and physiology through histology and single cell recordings. So it's a very useful testing ground to link some of those optical techniques to some of the known data we have from electrophysiology. We also know that techniques of visual psychophysics have been developed to quantify visual performance. So if we can record from the retina non-invasively in humans, we can also pair that in real time with behavioral experiments. And that's one of the powerful things of potentially using high resolution retinal imaging in conjunction with psychophysical experiments and behavioral testing. And we know a lot about how particular visual stimuli are processed. So we can stimulate or target particular subsections of the multiple parallel neural pathways that start in the retina. So is the retina a good testing ground for linking neural processing and behavior? So clearly the retina is a piece of neural tissue at the back of the eye. If we're thinking about imaging this piece of neural tissue, light would have to enter through the pupil, and the piece of neural tissue you may have seen in fundus images from the optician, for example. There are blood vessels running over the surface of it, so light has to pass through these blood vessels to the neural layers beyond. And there are layers of processing circuitry that are potentially accessible to us. So we have rod and cone photoreceptors. Their signals are fed in a hierarchy to bipolar cells and then to ganglion cells, and it's the axons of the ganglion cells that form the optic nerve. There are also lateral connections here. We've got either horizontal cells or amacrine cells that are forming lateral connections. And so this set of neurons, and there are many differentiated types of ganglion cell and bipolar cell, they form specialized circuitry for extracting information from the photoreceptor signals. So in a routine autofluorescence fundus image, you have images like this. And if you zoom in on that optically, there's no way that you can resolve those individual cells. So the tricks we need to use to get single cell resolution are, as we've heard about in other talks, using confocal imaging to obtain sectioning in depth. And the process we use there is to scan a point of light across the retina and then collect it in a confocal imaging system. But additionally to that, we need to measure and correct the optical distortions that are introduced by passing light through the eye's own optics to send this point of light onto the retina. And the way we do that is using an adaptive optics system. So we measure the reflected light and we send that to a wavefront sensor, measures the distortion, and we put the inverse distortion on a deformable mirror so that the corrected wavefront is sent to our detector. And so with that, we can obtain diffraction limited resolution. We're obviously influenced by the size of the pupil, but we've got diffraction limited imaging of the human eye, and it means we can get down to 1 or 2 microns, which is enough to be able to resolve even foveal cone cells and some rod cells as well, depending on the exact setup of the system and the wavelength that we're using. So this is our adaptive optics scanning laser ophthalmoscope in the lab. It's got the key components that I mentioned. So there's an original light source, superluminescent diode that we use. That's sent to the eye off some scanning mirrors. And in that path is a deformable mirror that we can use to correct the eye's aberrations. The stimulus gets sent to the observer's eye. We hold our observer still by asking them to bite on a bite bar. Light is reflected from the eye back through the path and to a photomultiplier tube or an APD. Clearly, we need the hardware in the lab to be able to do this, but you also need dedicated and skilled researchers, and I've got an excellent group of people who've been working on this project. So with adaptive optics, the types of images we get of the en face retina contain these little bright blob regions. So each one of these bright blobs is associated with a cone photoreceptor. And I'll talk a little bit more about this in detail, but the bright reflection is coming from the outer segments of the cones. So you can also see here, this is the shadow of a blood vessel, quite a small capillary here. I want to talk a little bit about what AOSLO allows us to do. So these are the types of images we get. We're measuring video frames at a rate of 30 Hertz. So this is a sort of real-time movie, actually, of one of my postdoc's eyes. It's on a loop, which is why it looks as though it's just going backwards and forwards all the time, but real-time 30 Hertz video of the cone photoreceptors. And typically what you see in papers is a stabilized, referenced, averaged image. And so what sort of information is available in data like this? Well, you can obviously extract cone statistics, packing arrangements, etc. We can use some imaging tricks to get at other structures other than just this really bright reflection from the cone outer segment. We can consider the retina in health and disease. We can identify different cone types. So you may know that there are 3 classes of cone in the retina, sensitive to short, middle, and long wavelength lights. And it's possible to identify those different types. We can also measure blood flow. And critically, we can measure eye movements, although they're a problem to us in capturing the data and analyzing it in terms of getting images of the cones. We can also, as a byproduct of doing all that, get really nice information about eye movements. Once we've done that, we can achieve single cone stimulation, and we can also think about extracting functional measures. So in the rest of the talk, I'll use these 8 examples and take you through some use cases for each of those. So very simple one, thinking about cone statistics. Well, if we image close to the fovea, we've got very densely packed cones. If we image at a slightly greater eccentricity, we've got more widely spaced cones. And in this image, the rods are below the resolution limit, but it's likely that there are rods that are interspersed in these gaps between cones here. We can also use these tricks of different ways of collecting the light. So most of what we're doing is confocal imaging, and that's where we're collecting bright signals from the cone outer segments. But it's also possible, for example, to block the confocal light and collect scattered light. And if you take the difference between light scattered on the left half of an annulus and the right half of an annulus, you get really nice data on the cone inner segments. So these images align perfectly with these. Each cone outer segment has its partner inner segment. And one of the reasons that this is nice is that often in pathologies, it's the cone outer segments that deteriorate. And if you were wanting to perhaps develop some retinal rehabilitation techniques, maybe gene therapy, etc., you'd want to know that there were still healthy inner segments underneath that. I think one thing that's always struck me about these types of data is that these are images that we're getting in the living human eye. And I think a rather wonderful parallel is the parallel between these in vivo images, where you've got split detector imaging of cone inner segments here. You can just see a couple of rods here, and that's beginning to get to the types of images that previously we've only been able to get in histology. So if you think about that in terms of monitoring disease, this is a study we did comparing healthy retinas and patients with an inherited retinal degeneration called Stargardt retina, Stargardt disease. And we can think about tracking disease progression on a very fine scale, but also setting up protocols that would allow us to monitor response to treatment, giving us potential endpoints in treatment trials. And as I'll discuss a bit later, linking structure and function at the cell level. So I also said that we can potentially identify different cone types. So these are some data from Hoffer, Singer and Williams, actually back in 2005, when imaging the photoreceptors like this was just possible. And they used a technique called densitometry to selectively bleach the different classes of cone using different bleaching wavelengths, and then compare reflectivity before and after the bleach. And if you do that enough times with the right combination of wavelengths, you can identify the short, middle and long wavelength sensitive cones. And there were other data that told us about this, which is that in the normal population, there's a massive variation in the ratio of particularly L to M cones. But all of these individuals have normal color vision. And so there are compensatory neural mechanisms that compensate for the fact that this retina gives you obviously a large L signal and not so much M signal. And taking that a little bit further, so classifying the cone types in normal retina and in dichromat retina. So these are individuals who are red-green colorblind. This individual is lacking L cones. This individual is lacking M cones. They're lacking those cone classes in slightly different ways. In this case, there are no L cones existing at all. Here, the cones are existing, but they don't have functional waveguiding properties, and so they don't give us bright reflections. So beyond the photoreceptor mosaic, what else can we do? We've got confocal imaging, so we can section in depth to some extent. Moving to a different depth plane, we can image blood vessels, and before that, we can image the nerve fiber layer. So again, thinking about neural degeneration, imaging the nerve fiber layer is really important. But with blood vessels, we can begin to image blood flow in real time. And we can do this in some of the smallest capillaries in the retina, and it gives us a direct, non-invasive assessment of localized retinal blood flow. One of the big questions in neuroscience, I think, and particularly because of some of the technologies that are used to understand neural function, is the relationship between blood flow and neural activity. And so here we've got a potential testing ground for thinking a lot about neurovascular coupling. So here's an example from Steve Burns' lab. Again, this was right at the beginning of being able to use these techniques. There are more sophisticated experiments done now, but this was one of the first demonstrations. So there's a measurement site here, and we're plotting blood velocity here. And then this is a stimulus that's presented. So this stimulus is obviously going to generate neural activity, and we look for the modulation in blood velocity. So there's a control condition and a condition in the lower part of the retina here, which will be driving blood demand through these vascular networks, whereas either stimulation across the whole area or just in the upper retinal area will be driving blood flow in these areas, and that's what we see in the data. So one of the things I've been particularly interested in is actually the byproduct of extracting very fine information about eye movements from AOSLO data. So this is an example of a single frame, and in this single frame that we've captured, the eye hasn't made much movement, but in this frame, the eye's made a lateral movement during stimulus capture of this frame. Because it's a scanning system, we get very systematic distortions. We can image process and analyze these distortions to get information about how the eye has moved. So this is a typical eye tracker recording at the same time as our AOSLO recording. And just thinking about the information that we get from the typical video-based eye tracker, plotting XY position here, we can see that when we're doing that with the retinal imaging data, we've got much higher fidelity, and we're recovering information about specific microfixations that people are making. And so one of the questions we've been asking in the lab is thinking about these different types of individuals who have different LM ratios, and then devising a stimulus that is identified either only by the M cones or by the L cones. And it's a stimulus that's sufficiently small that puts us in a sampling limited regime in terms of identifying the gap in this C-shaped stimulus. It's a behavioral task, and we ask, depending on your L to M ratio, are your eye movements adaptive to this task? And we're asking specifically about the way in which the photoreceptor mosaic samples information. So it's a very active process. Now, once we can measure eye movements, if we can do it in real time, we have the potential to be able to update the stimulus to track a particular location in the retina. And this is a movie from Austin Roeder's lab. So we have techniques now, real-time techniques that allow us to track the retina over time and to modulate the light stimulus that we're presenting to track a particular cone and to stimulate that particular cone. So the way we do this is we use an acousto-optic modulator that's coupled into our eye movement extraction. And we typically use one beam for scanning and imaging, and another beam, say a 543 nanometer laser, to give a stimulus at a particular location. And so once it's possible to do that, it's possible to ask about specific cone stimulations. And this is a case where Harmening and Sinčić have assessed psychophysical thresholds for stimuli that are either presented to a cone or to the gap between cones. So they've got successive trials in their experiment, and the y-axis here is slightly confusingly labeled, but it's basically the intensity of the stimulus. And when the participant gets it right, the stimulus is made dimmer. When they get it wrong, the stimulus is made brighter. And these so-called staircases should home in on a threshold value. And so over a number of cones and gaps that they tested, you get nicely significant differences between thresholds for light that falls on a cone and light that falls in a gap, which is as you'd hope, but this is really rather a neat demonstration of the capability of the technology to now stimulate individual cones in the human eye. Now, I guess the holy grail in this is to be able to directly visualize neural response. And so one of the things that people have been searching for is an intrinsic change in these AOSLO images that comes as a result of neural response. And the first level of neurons that have been looked at are the photoreceptors. So by stimulating the photoreceptors and then looking at reflectance over time, so we stimulate with a 550 nanometer stimulus, and we look at standardized reflectance as a function of time after stimulus onset. And typically some cones will get brighter, some will get dimmer, and some won't have much change at all. But if you aggregate over populations, you can look at the standard deviations in the response, and stimulated regions show large variation in the change in reflectivity. And so that's been used to nicely map out the action spectrum of this signal, this reflectance signal. And in 5 different subjects here, we're doing that at different wavelengths, looking at stimulus intensity, and then measuring off a particular threshold stimulus intensity, a threshold amplitude change, and finding the stimulus intensity and plotting out the stimulus intensity needed as a function of wavelength. And these data for these different wavelengths, averaged across the 5 people, are plotted on top of the luminous efficiency function of the human eye. So this nicely links these reflectance changes to the spectral sensitivity of the L and M cone photoreceptors. Now, most mileage in this area has come from interferometric techniques, as you might expect. Just over the last couple of years, couple of months, things are coming out that are really exciting in this area. So if we use optical coherence tomography, which is an interferometric technique, also coupled with adaptive optics imaging, you can also do this at the same time as scanning laser ophthalmoscopy. You can get much better resolution in depth. And so we extract these 3D volumes. And there are 2 bright reflections here, one that are basically both coupled to the outer segment of the cones, but one bright reflection comes from the inner segment, outer segment junction, and one comes from the tip of the cone outer segment. And in response to light, these 2 surfaces move apart. And so there's an optical path length difference that is associated with conformational changes in the photopigment molecules that is directly associated to the amount of pigment that is bleached. And so there are 2 components to this response that have been measured so far. One, a very short component of the order of milliseconds, and a much longer component here. This one is thought to be the early receptor potential, and this is thought to be a mixture of the late receptor potential and some osmotic changes. And so using that change in optical path length, Zhang et al. Have mapped out the cone types of normal trichromat retinas and deuteranote retinas. And this is different from the data I was showing you before, in that this is real time in terms of the activity of those cone cells in response to these 3 types of light. And so just to wrap up, I want to think about inspirational data, I guess, and looking forward to where this might be able to go. So this is a wonderful OCT movie from Lang et al. I've talked a little bit about measuring the functional response, intrinsic functional response in the cone photoreceptors. In a way, what we'd really want to do is measure the intrinsic functional response in retinal ganglion cells. These are perhaps more interesting in terms of neural processing, neural circuitry, etc. But one of the difficulties of imaging the ganglion cells is that light typically has to go through the ganglion cell layers to get to the photoreceptors, and they're quite transparent. But again, using interferometric techniques and OCT, this is a movie going through the nerve fiber layer, and then these are images of ganglion cells, and then through to the inner nuclear layer. And you can't see it very easily, but that's the ganglion cell layer, and coming down here, they're now much smaller than the ganglion cells. You get to the cone photoreceptors that I was talking about in the other images. Now, if we could couple that with actually being able to measure activity in these ganglion cells, that would be wonderful. So this is in vitro recording of deformations associated with action potential spikes. These are on a much smaller scale, just a couple of nanometers compared to the 100 nanometer changes we were getting in the photoreceptor outer segment length, but potentially really exciting for measuring function in the retina. So powerful tool for investigating vision. We've got structural imaging of cone photoreceptors, cellular level imaging of retinal blood flow, opens up exciting avenues in neurovascular coupling, eye movements and the way that the visual scene is sampled at the level of the photoreceptor mosaic, single cone stimulation, and then the holy grail of intrinsic optical signals of neural function, allowing direct real-time measurements of responses of hundreds of cells in parallel in the living human eye. Thank you.
That's great, Hannah. Thanks so much. Really great talk. There is a question in the Q&A text feed, which it'd be great if you could address offline. But in the interest of time, I think we'll move on. And I'd like to invite Parashkesh Nashev from University College London to give his talk entitled Mapping Complex Causal Fields in the Focally Injured Human Brain.
So the subject of this talk is how we address the following very common problem, which is given injury to the brain, focal injury, most commonly the kind that occurs in stroke, how can we predict what happens to the patient who suffers it? It's inevitably a process of causing an interruption of various patterns of interacting functions within the brain. And what is very striking about those circumstances is that the outcomes here are highly diverse and individually very difficult to predict. Now we can recast this problem as the transformation of a set of state of affairs into another, where the causation is distributed across a wide array of factors, what we might call a causal field, that is here being disrupted. And so the question is, what is the space of possible solutions here? Well, one possibility might be that the causal field is relatively small. Although the brain might look very complex, the actual underlying causation is simple. Or it could be that there is a large underlying causal field. Now, these two possible alternative categories of solution clearly have different types of associated models. Let's imagine that the underlying causal field is small. Well, that is precisely the kind of circumstances we would like to observe, because then our models can be simple, they can be elegant, they can be parsimonious, and they can be easily intelligible. But it's also possible that the causal field is large, and that would require models that are the exact opposite. So what can we say about the macroscopic function organization of the brain? Well, those of you who study this topic will know that fairly characteristic pattern tends to emerge. Here what I'm showing you is a graphical, a generative graphical model of the brain with a focus on the structures that are engaged in the operation of the autonomic brain. So what we've done here is to build a model of every point in the brain where each voxel is treated as a graph, as the node of a graph, and then each edge is estimated from an array of connectivities, white matter connectivity, functional connectivity from resting state, functional imaging, and patterns of change in gray matter. And this is a stochastic block model, which gives us an indication of what is the underlying community structure of patterns of connectivity. And what you see is, yes, there are some broad patterns that we can observe that appear to be relatively simple, but as one moves to more finely granular description, the patterns become more and more complex. It's not only the underlying macroscopic organization that's complex, it's also true that the causes of focal brain injury are complex themselves, and this is to be expected. When we think about it, the way in which a stroke lesion presents is likely to be influenced, on the one hand, by the nature of the vascular tree, whose occlusion is its cause, by the pattern of occlusion that occurs, and of course, whether or not a particular lesion produces a symptom, what neurologists call symptomatic eloquence. And what we see is the outcome of those three fairly complex mechanisms operating in tandem. So now if we look at the spatial patterns of individual stroke lesions, we find that they're certainly not random. They have a structure, and they have a structure that is neither completely scattered, nor is it very simple to describe. Here I'll just zoom in, and if you can see, oops, as I zoom into this plot. So this is a two-dimensional embedding of a set of stroke lesions. These are actually hemorrhages that have been projected from the side. So you can see these little mini brains, and there is a cluster here because these are all hemorrhages in the cerebellum. And as we zoom out, we can see that there are other clusters, some of them larger. Here we see the usual number, perhaps the most commonest form of deeper hemorrhages. Up here, there's some cortical ones, and so on. So the point here is that the patterns are neither completely chaotic, neither completely random rather, nor are they simple. And the same kinds of appearances can be seen when we look at ischemic lesions. So here what we've done is to embed 1,300 lesions within a two-dimensional space, and then derived archetypal clusters of characteristic patterns of damage. We can extract a reasonably compact representation, but even if we do so, our ability to predict what happens to a patient based on that kind of simple redescription remains relatively weak. And now let's stand back and think about the much wider domain of potential causes of damage to the brain. What you're seeing here is a representation of how radiologists see the brain. What we've done is to analyze 100,000 reports of MR scans across the full array of possible pathological conditions. And here each report is represented in a two-dimensional space based on the characteristic pattern of asserted pathological changes and anatomical locations. And it's labeled by the patient's age as a color map on the side, which is not part of the report. So you can see what we observe is a complex clustering of multiple characteristic forms of appearances as the radiologist sees them, that are here predominantly in the aged, which tends to be patients with neurodegenerative disorders. Here, these younger populations tend to be inflammatory, and the mixed here are neoplastic, and so on and so on. So the patterns of damage here become even more complex and even less plausibly modeled in any simple way. And then we need to consider why it is that the relationship between damage to the brain and its impact on the patient's function is actually so hard to do, what you might call lesion deficit inference. Well, the situation is wholly unlike the case where we're mapping the function of the brain with something like functional imaging. This is represented on the left here. We have the distributed neural substrate, the substance of the brain generating behavior or supporting behavior, and in parallel it also activates the brain as recognized or as measurable through bold changes across individual voxels that one can then serially and severally study. And so mass univariate inference that is familiar to all of us who do functional imaging is perfectly licit here, at least for the purpose of mapping patterns of functional activation. But when we look at the case of lesion deficit inference, what we have is a lesion composed of multiplicity of areas of the brain that are damaged, itself driven by complex pattern, complex pathological pattern, that then causes a deficit through the way in which it disturbs the underlying distributed neural substrate. So the relationship here is causal and by definition distributed. So it is not possible for us to take each individual voxel and analyze it more or less in isolation in the way in which is perfectly reasonable within the functional imaging domain. So what are the consequences of this complexity? Well, let me demonstrate it through a very simple simulation. So here we have a simulated brain. Well, that happens to be a two-dimensional square with the functionally significant area being a question mark. This is our unobserved functional substrate. And then we have a set of observed lesions and observed behavior. What we're trying to do is to infer what that underlying functional dependence is. Now note that here there is no spatial ground truth. We don't know exactly what parts of the brain are responsible for the function that we're disrupting. That is exactly what we're trying to find out. So first, let's imagine that we have a set of large lesions. Here are some examples from the simulation that is just done in this very simple pixel space. Here's a lesion distribution of about 1,000 of them. And then if we do a conventional form of inference of the kind that we would do in functional imaging, this is what we extract. Yes, we can discern the overall structure, but you can see it's all very crude. And this is within a relatively, obviously it's a synthetic, noise-free environment. If the lesions are small, however, then we're able to produce a better retrieval. And so there's an obvious relation between the size of the lesion that we use in order to do our mapping and the fidelity of the final outcome. But let's now imagine that what is changing is not merely the size of the lesion, but its spatial organization. That we have a lesion distribution where each lesion has a characteristic structure of the kind that I've shown you exists in reality. Here the lesion distribution ends up looking different compared with when there is no such bias. But crucially, when we now try to do inference, we end up with a very distorted picture. So here these lesions have been organized in the shape of a vascular tree to simulate a kind of vascular tree. And so certain areas now appear to be much more significant than they really are, and other areas are neglected. Now you might say, well, that's simply a simulation. It does not mean that that has to correspond to any kind of reality. But we can do exactly the same simulation now based on real lesion data, where we posit a set of ground truths, and then we see what is the displacement of the actual real ground truth compared with the estimate. And given a set of single voxel ground truths, we get these sorts of patterns of displacement here indicated by vectors pointing in the direction of the displacement. And you can see that you end up with a beautiful map of the vascular tree of the brain. Here's the territory of the MCA, here's the anterior cerebral, here's posterior circulation. And the degree of displacement is very substantial. It's of the order of 10 millimeters or more, enough to cross boundaries between brain lobes. Now imagine that spatial dependence is not a single voxel, which would be implausible anyway, but combinations of larger areas. Here, for example, Brodmann area 44 and Brodmann area 39, two candidate areas for the locus visual spatial neglect. And we end up with the inference being displaced into a completely different region, superior temporal gyrus. Does this mean that there is no solution? Well, no. If we're capable of capturing the relationships between the various patterns of focal brain damage and disentangling them from the underlying functional organization, which we can do with high-dimensional multivariate methods, then we can produce a much better result. We're still limited by the data, but much less so. And if we transfer our simulations to the brain, we find that under simulated ground truths that are plausible, we end up with better retrieval. So we do need to consider how we approach the problem where the causal field is large, where the dependence between outcomes in the brain and damage to it in a focal pattern is highly complex. And what that will inevitably require is much more complex models than we're accustomed to, and everything else that goes with it. If we're using these more complex, flexible architectures, we'll obviously need large-scale compute. And most importantly, and perhaps hardest to obtain, we will need large-scale data. How do we do that? Well, luckily, rapid improvements are being made in the quality of imaging that we can actually deploy at the clinical front line. Recently, a collaborator of mine at Chiao Tung University has developed a remarkably fast 6-minute method for imaging the brain high resolution with sensitivity to metabolic changes, such as the lactate that tends to accumulate under ischemia. So we can produce these beautiful maps of ischemia in a clinical context. At a rather earlier stage of development, there's a new method arising from geophysics at Imperial, Mike Warner's group there, that should promise to allow us to extract almost an MR quality image from an adult human brain purely with ultrasound. This is now not using tomography, which will only produce a blurred image, but using a much more faithful rendering of the physics for wave inversion. But these technological advances aside, and the feasibility of more complex modeling, we should perhaps think about sets of objections that one might have to the very idea. After all, we dislike having to operate in the realm of models that are so complex that they become opaque. And so we should consider some objections. The first and very common one is that surely biological causality must be simple. It's attractive to expect the brain to have to operate with relative simplicity. Well, I have to say that I think there is no reason for us to be sure of that. It may well be that it's complex. In fact, complexity is surely a necessity to maintain the flexibility the brain needs. It's also commonly assumed that biological causality has to be generalizable, that what we study are general rules that must be common across each instance of the brain. Well, we have to remember that biology does not and probably cannot care about overfitting when it comes to the solution to the biological problem that each human being represents. And in particular, when we consider the difference between the maximal genomic content, here represented in terms of equal areas, and the minimal brain information content. One is assuming all of the genome is coding, the other is assuming that the brain is essentially composed of neurons that operate as simple logic gates. Disparity between the two is huge. And so that gap can only be bridged in a way that need not generalize across the population. And then finally, of course, the premise of all science is that what we're doing is intelligible. But biology is under no pressure to be intelligible to us. It just has to work. It can stay as opaque as it likes. And so we needn't be resistant to the use of models that are opaque, that are difficult to understand, but nonetheless happen to work. Because ultimately what we have to remember, or at least those of us with clinical responsibilities are forced to remember every day, is that the focus of medicine is always the individual patient. It's the person in front of us. And the priority for any of the modeling we do is that we should be able to apply it as faithfully as possible to an individual patient. So I'll just leave with one final large causal field, which is the members of my group and the many collaborators that have made all this possible. Thank you very much. I'll stop there.
That was lovely, Parashkev. I may have missed it because I was doing some sort of chair management issues in the background there, but in your work, are you taking into account the tissue type, you know, white matter versus grey matter and things like that in these causal fields?
Yes, I mean, that's definitely an important way of compressing the signal, because clearly white matter disconnects grey matter, and an attractive way of representing damage is to use a disconnector, to use a representation of white matter damage in terms of the grey matter that it disconnects. I think finding optimal representations of brain damage is going to be one of the central tasks for us. How can we come up with a compact, succinct, navigable representation of those patterns so that we can then combine it with the even greater complexity that we see of the macroscopic organization of the brain.
That's great. Thanks so much. I think we've got plenty of material from your talk and others for the panel discussion. We've got one more talk to come. So I'd like to invite Allard Robroek from Maastricht University to give his talk. And while he's just preparing, I should also plug the York Maastricht Partnership, which is something that University of York and Maastricht University has established in recent years. And one of the areas in which we collaborate is in neuroimaging. So I've met Alaa before and welcome him here today. He's going to give the talk entitled Multi-Scale Investigation of Human Cortical Architecture with Light Sheet Microscopy and MRI. Over to you, Alaa. Thanks for coming.
Thank you very much, Tony. I hope everyone can hear me, see me, and especially see my slides, so that all the sharing went well. A pleasure to be here. Sorry to be giving the last talk. I hope I can capture your attention for a little bit more time. There we go. What I want to talk about today is imaging the human brain, and in particular imaging the layers of the cortex and imaging its connectivity. And of course, one of the characteristics of the brain that gives you most pain in trying to image it is the many scales at which it's organized. And, you know, just very quickly, something that's familiar to many of you, particularly to those of you that work with the brain and imaging the brain, we can consider it at many different levels: at the macro scale, the mesoscale of the cortical layers, the micro scale of the neurons, and even the nanoscale of synapses. And so there are also typically different modalities of imaging the brain that work best at one or perhaps two of these scales. In particular, today, I'll talk about ex vivo magnetic resonance imaging, so magnetic resonance imaging of the human brain in the postmortem state, and ex vivo light microscopy that tend to straddle the mesoscale and the microscale of layers and neurons. And the obvious trade-off that you will have between the resolution of the imaging and the field of view of the imaging. Now, after having introduced that, let me start with the MRI today, and then later get to the light microscopy. MRI resolution typically on clinical 3 Tesla scanners will happen at resolutions on the order of 1 millimeter or 2 millimeter isotropic, sometimes anisotropic by that order of magnitude. And clearly, if you look at the layered structure of the human neocortex, that isn't nearly enough to capture the structure that's there. You would have to go to at least 1/3 of a millimeter isotropic in order to cover that well. Now, we do that in MRI here in my lab in Maastricht using very powerful human-sized bore 9.4 Tesla and 7 Tesla scanners. In addition to specifically built radio frequency coils that are indeed conformal to postmortem human cadaver brains in order to get to the kind of resolutions that we want. And in addition, we scan for a very long time, which is a benefit that you have when scanning ex vivo brains rather than in vivo subjects. And also then we have to crunch terabytes of data afterwards in order to reconstruct the images that you're going to see. So here you see one of the earliest images that we did quite a number of years ago using that machinery, a human brain. Here you see an actual section, an axial section through a T2 star weighted image. I hope you can appreciate the whole brain field of view coverage, the exquisite contrast between white matter and gray matter. And also if we zoom in a bit further into the cut through the hippocampus, we can actually have resolution at 200 micron isotropic to start to see the layering of the hippocampus. And so resolution there really helps. In addition, I hope you can also appreciate on your screen at home, contrast also does a lot in the sense that in the white matter even, you see a nice contrast, a T2 star contrast to distinguish one white matter tract from another. We didn't stop there though. We went right on to trying to achieve 100 micron isotropic anatomical imaging of the human brain. And I'm showing you some data here. Again, this was a whole brain data set, but right now I'm zooming into a very famous piece of human neocortex, the primary visual area in the calcarine sulcus, V1, which is of course well known to be characterized by the stripe of Gennari, the SOG. And we were very pleased to see that at this level of imaging, not only did we have the field of view to cover the entire brain, but also now we had the resolution and the contrast to actually see and distinguish the stripe of Gennari throughout V1, and therefore to anatomically determine in these scans where we could find V1. Now, we didn't stop there. The data set with the highest resolution that we've done to date of an entire human cadaver brain was 75 micron isotropic. Initially, it doesn't sound like much to go from 100 to 75, but it is indeed about double the resolution and also double the imaging time. Here you see some sagittal, coronal, transverse cuts to the entire T2 star-weighted data set. I hope you can appreciate the whole brain coverage and the resolution, not only between white matter and gray matter, but also between subcortical structures, and myelination, but also iron content has a lot to do with creating that contrast. But zooming in a bit, in particular to this bit over here, into the cortex, I hope you can appreciate on your screen that we have the contrast and the resolution here to actually start to distinguish multiple individual layers in the human cortex. And in particular, you can really distinguish the two layers of Baillargeau, the inner and outer layer of Baillargeau, that are very characteristic of bistriate myeloarchitecture in certain areas. Now, we didn't stop just at the myeloarchitecture of cortex. One of the particular aims of my group, like I said before, is to look at the connectivity of the brain. And in magnetic resonance imaging, that means often looking at diffusion MRI. It's a very common use modality in vivo, but we've developed it in particular for ex vivo. And here you see some result of processing of diffusion MRI data, where if we take this rectangle here of the coronal cut through the brain, where we also got the very high resolution structural data, or well, scalar T1 weighted and T2 weighted data. Here's a zoom in to a diffusion weighted volume that we did, where you can appreciate the large connections that are well known, like the corpus callosum or the corticospinal tracts, and that are visible at resolutions of 1 to 2 millimeter isotropic in vivo. But at 400 micron isotropic, we can also start to appreciate much more structures, such as fiber tracts curving into the cortex and actual intracortical structures. And here on another image, you can see that a bit clearer. Here you have a so-called tractogram, which is tractography performed on all of those individually analyzed directions from the diffusion data, which can be formed to actually can be used to actually form macro scale connectomes. But at 400 micron resolution, we actually have the resolution to start to trace some of these connections into the cortex, also at the point where they tend to bend very sharply into the bank of sulci in particular, which are things that you often miss when you perform this tractography at lower resolutions that are feasible in vivo. And so in that sense, we can not only look at the cytoarchitecture, or especially myeloarchitecture, but also at what you could call the connect architecture of cortical columns of the human cortex, often, of course, in modern terms called connectomics. Now, that brings me to light sheet microscopy as another modality that our group has used in order to look at cortical architecture. And obviously, if you talk again about the cortex and its basic size and the resolution of the structures in it, once we look at one of these 1/3 of a millimeter isotropic voxels that we were aiming as the target of resolution in MRI before, then basically where it comes to light microscopy, that would be a common field of view that would be used just 5 or 10 years ago, or in fact, in many common labs nowadays, because that's the basic field of views that you can reach with confocal microscopy and 2-photon microscopy. Now, our aim for light microscopy was really to try and straddle the field of views and the resolutions of the MRI that we were doing and the light microscopy that we were doing. And so the basic aim was to maintain the micrometer resolution of light microscopy and fluorescence microscopy, but to enlarge strongly the field of view. And in order to do that, we could make use of a development in fluorescence microscopy that's called light sheet microscopy that fundamentally alters the way in which you go through a sample, whereas confocal and 2-photon microscopy point scan their way through the sample, causing quite a bit of bleaching of the label outside of the focus, but also especially even with modern resonance scanners, it gets you through your data kind of slow, and therefore the coverage that you can have, the field of view that you can have, is limited. Light sheet microscopy will project a very thin sheet of laser light through the sample, and will immediately, with a CMOS camera, get an entire plane out of a sample. And then by moving the planes along through your sample, you can get data sets such as the one that you're looking at now, Here you see a scale bar of about 200 microns. Here you see the depth of the imaging, how far we're going down with the plane. And I hope you can appreciate that with this cell body stain into the cortex, you see some vascular structures, but especially what you see is the structures of neurons. You can now and then see some structure of microcolumns. You can see layer 1, the neuron sparse layer of the cortex coming in, and you can entirely go through your sample very fast and over large field of view. Now, our aim was to apply that to large pieces of human neocortex, such as this, of a couple of centimeters and a couple of millimeters thick. But in order to actually apply light sheet microscopy, you have to actually make your tissue transparent. And so you actually have to get really good at something that's called tissue clearing, optical tissue clearing. And so that's what we set about to do. And we developed, and especially Sven Hildebrandt in my lab, developed a method called MASH, multi-scale architectonic staining of human cortex, where there is quite an interaction between different steps into the entire chemical process of optical clearing and labeling and making it suitable for light sheet microscopy. And so in particular, in the labeling, we developed some new small molecule dyes, and in the refractive index matching, which is also crucial before you can image, we developed some new refractive index matching solution. And all of this led to clearing and labeling very large samples such as this in about 10 days for 5 millimeter thick samples. Now 10 days might sound like a lot, but that was a lot more before we came along. And it's a very acceptable thing if you think about the fact that if you want to have a biopsy with this size or a resection from a surgery, and normally you take thin sections and do 2D microscopy on each of those, it would take you many months. So that's quite a nice time frame to reach there. And also importantly, we could keep the dye costs to about $1, £1, €1, whatever have you, per sample, which is also, if you consider that to stain this entire block, even if penetration would allow with antibodies, would cost many hundreds or thousands of euros or pounds. Now, just a little bit more about the dyes. There are fluorescent small molecule dyes, which are very fast diffusing, which means they can actually get into samples as large as this, whereas large antibody molecules cannot. And we developed 4 dyes into different spectral bands that either label cell bodies or cell nuclei, and are therefore very, very suitable to look at cytoarchitecture in the cortex. So here's an example from the paper that we published last year about how we try to put everything together here, how we really try to image the human brain and its cortical architecture at multiple scales. We first performed MRI on a human occipital lobe. Here you see a right human occipital lobe, still a piece of the splenium of the corpus callosum. This is the parietal side. Here's the calcarine sulcus. And so here would be V1. And indeed, you can distinguish, if you look carefully, and I hope you have the resolution at home, you can distinguish the stripe of Gennari. And so here V2 would start, both dorsally and ventrally. Now, after having done MRI on this, we actually excised pieces of tissue and cleared them in the way described before. And the aim was really to image from the brain down to the neuron via the cortical layers. And so we used the light sheet microscope and zoomed in a little bit further. Here you see a combined MASH MG and MASH NR stain, so a dual color stain for cell bodies and cell nuclei, where indeed in V1 and V2, with this particular piece over here, which would be located about over here, you can see the nice layering in the cortex. And then zooming in even further at the field of view over there, you can see that light sheet microscopy imaging like that is actually suitable to do the complete cytoarchitectonic characterization of the cortex. Now, taking this a bit more anterior to a 5 millimeter thick block, just a bit anterior over there, and then looking at V2, again, you can see the beautiful cytoarchitectonic layering, the characteristic large pyramidal cells in layer 3B. And I can actually show you a running movie of that, where once again, you see the relatively raw light sheet microscopy data set. While, by the way, I think my son is starting his drumming lessons over there, so I hope that doesn't disturb the sound too much, but I'll keep continuing. You can see how the light sheet passes through all of cortex, how you can go millimeters deep, and also how really you can distinguish the large pyramidal cells of layer 3, the granular cells of layer 4, and of course the cell sparse layer 1. Zooming in a bit more, you can actually start to see microcolumnar structures of neuron cell bodies. And in fact, the resolution of this data was enough, and don't forget the fact that it's 3D, to start to segment single neuronal cell bodies. So here you see a single pyramidal cell body segmented in 3D from data sets like this. And so I really think we're getting close to imaging multi-scale from the brain down to the single neuron. Now that brings me to the outlook and the overview. I think we are getting close to better tools to investigate the human cortical architecture, the cyto, the myelo, and the connect architecture on the same human brain. Where it comes to MRI, we have a whole brain myelo architecture-like contrast in the T2 star in 3D and over the entire brain. And the aim is to correlate and validate that with light sheet microscopy on the same sample. And then where it comes to light sheet microscopy, where we've recently been advancing the methods we developed a lot, we really want to scale further the imaging component after having really tackled the clearing component and the labeling component. We want to scale the imaging component through a new light sheet microscope, a so-called CT-DSPIM, a cleared tissue double selective plane illumination microscope, which allows very large samples to be housed underneath it, and then the microscope to scan over those, and then by many stripes of high resolution imaging, you can actually get a stitched mosaic image of very large human postmortem brain samples. You see a small pilot that we did just 2 months ago over here at about 16 micron isotropic, and then stitched to about 4 by 8 centimeter by 5 millimeter thick. We're also still looking at new small molecule labels, where we already have the MASH cyto labels. We really would like to create some myelin labels and also labels for blood vessels. That is also because blood vessels are, of course, the basis for functional magnetic resonance imaging and give another potential for some cross-linking and multi-scale investigation of the brain. And then finally, we do need to invest a lot, just like many people need to do, and especially many people that are in one of the omics, into storing, loading, visualizing, and also not immune to starting to use deep neural nets for identifying, counting, and classifying neurons and layers. Now, with that, I want to thank all my collaborators, especially my Maastricht team that has in recent years, especially Frank and Arco and Robert, worked on the MRI that I've been showing you, and Sven and Anna working on the light sheet microscopy that has been showing you, and my collaborators and my grants, and you for your attention. Thank you very much.
Thanks so much, Allard. That was a wonderful talk. I had a quick question, I guess, which is you use ex vivo tissue for the MRI, and clearly there's a signal to noise issue there in terms of you scanning for 60 to 80 hours. What does the future have in store for the living brain in terms of ultra high resolution?
I think a lot. That's definitely something that my group is also doing, and a few more groups in Maastricht, I think also in New York, are doing. One of the aims that I've always had with doing the ex vivo imaging, and you're right, there's a lot of problems you solve ex vivo that actually make it harder to image ex vivo tissue, such as the shorter T2s, the reduced diffusion coefficients. So you actually do struggle to get higher resolution than in vivo. But then when you work hard, you get there. But the aims in doing this, one of the aims has been also to see what resolution would be needed in vivo in order to see structures like columns, layers, microcolumns. That looks to be definitely below the 500 micron scale. And I have to say that I find the developments there promising in the sense that we are getting very close to that and surpassing that in a number of MRI modalities, like for instance at 7T and 9.4T in vivo human, we can now do T2 star imaging at very close to 500 and slightly below 500 micron isotropic. So we are starting to get there. Yeah.
Fantastic news. Great. Well, thanks so much for your talk, Allard. I think if you stay on video and if the other speakers for the session could join us on video, we can have a panel discussion. So Amanda, David, Hannah, Parashkev, if you could join us, that'd be absolutely terrific. And any other speakers or indeed folks who have been chairing or anything like that, that would be, I'd welcome you too if you're happy or willing to come. I thought that was a lovely session, and a few sort of things. I just three things sort of came to me. One, what we're measuring, what we can measure, the scale of those measurements, and then how can they interrelate? Because Allard is bridging two scales effectively. But I guess my fundamental thought was that we don't want to be siloed in being microscopic. In a sense, the true value of some measurements will be how they can bridge gaps and really allow us to build on our understanding. So perhaps I'd like to listen to your thoughts on that sort of issue to start off with. Parashkev, you've always got something useful to say, and you're a proper doctor, so you could tell us something really helpful.
I think the critical question is knowing how to select the scale that happens to be informative, and one has to do some kind of selection, because the moment you descend, I know we have trouble modeling 1 millimeter scans, let alone 75 microns when we're trying to be whole brain. Because the kind of model that we need to run at 1 millimeter becomes infeasibly large. So I think my suspicion is that the right solution would be to have some kind of hierarchical representation where you can gracefully clip the detail of your representation dependent on how much data you've got, and of course what you need for the problem that you're trying to solve. I mean, the clinical perspective is, of course, you're always interested in the utility of what you're doing, and your ultimate measure is useful or not useful. And so the model might end up being not quite as elegant as you would like it. It's less of a problem than that it might not work very well in a patient group. One of the horrible things about being a clinician is you cannot say to a patient, I'm sorry, your noise, you don't fit the model, because every single patient counts. So I think my suggestion would be hierarchical representations that are then adapted dependent on the task.
If I can react to that, I immediately had two thoughts to what you said, Parashkev, and I fully agree with you. On not being able to do 75 microns, well, obviously I've been able to do it once on a cadaver brain, and so we should put that into perspective. But certainly in MRI, and I also thought I saw that in your talk, what contrast you choose greatly determines your resolution. Like for instance, even ex vivo in those cadaver brains, I could achieve 75 microns in T2 star weighted imaging, but only 400 microns in diffusion weighted imaging. And I worked quite a bit harder for that 400 microns than I did for the 75, just because the diffusion weighted modality is so much harder to get to, the signal to noise there, than the T2 star weighted modality. However, the diffusion weighted modality gives me much more information per voxel. It gives me entire directions of where fibers are traveling. And even if you push a bit more, you can also learn more in the contrast in every single voxel between intracellular processes and extracellular ones. So I think resolution and contrast interacts there. So that's my first thought. My second thought is I fully and entirely attach to your idea of hierarchical representation and hierarchical imaging. And that's exactly how we are approaching the very large tissue light sheet right now. So we work hard to do 16 micron overview scans of entire pieces of tissue that can be 5 centimeters by 5 centimeters by 5 millimeters, to use those to zoom in and go and do 4 micron isotropic, to use those and zoom in and go to 1 micron isotropic. And it's really for us now, and I think into the future, the only way in which you can do imaging like that, like short of doing 1 micron of the entire sample. So yeah, definitely fully agree.
Yeah, I mean, it'd be wonderful, of course, to be able to operate at much larger scale than we do. I mean, it would be great if every single patient had, half a millimeter or 100 micron scans. I'm sure we would make use of them. What I think we're missing is, on the clinical side, is the infrastructure that would render that kind of imaging modelable. It's always seemed to me a scandal that a country such as the United Kingdom has a supercomputer for predicting the weather that costs 100 million, and yet we do not have any supercomputing facilities provided for the NHS. I mean, not even for storage, let alone for compute. And so it would be, I think, very important for us to make a case for gathering that kind of rich data in the clinical population, and then that would then allow us to motivate the building of the necessary infrastructure in order to be able to use it properly.
Prashkev, I'd be interested in your insights on sort of prospective evaluation of the brain, rather than, I mean, you tend to pick up, you know, the catastrophic failure of the brain through a stroke or something like that. But, you know, what does the future hold for, potential screening or measures that would be of value to predict what the future might be for that patient's neurology?
Well, yes, I suppose the problem with imaging in a clinical context is that we only image those in whom we suspect there may be something wrong. And so our data collection is, by definition, very heavily biased. It would be great if we actually had an MR image on every single person in the country. I mean, if I could, I would definitely set up that study. That would be wonderful. And it would be, I think, only then that we will be able to comment a bit more on what the longitudinal change might be and to build the right kind of predictive analytics. The other thing, of course, is that we tend to speak of predictive models when we apply machine learning in the realm of neuroimaging and medicine in general. But what we really want is prescriptive models. We want a model that tells you should do X in this patient or Y because this is what you want to achieve. But of course, prescription implies a counterfactual, implies a prediction about an individual given an action, and building counterfactual models in the context of highly biased data collection is really hard. And that, again, just needs more data and more of the right kind of data.
So
There's a big political piece to do that.
I wonder, Prashkev, when you say predictive modeling, where you would like to use an MRI scan of every single human in the country, when you look at initiative like the UK Biobank, and well, they're not getting everyone in the country, but they're getting 10,000 to 100,000 subjects. How far does that go towards that goal for you?
Well, I mean, it's definitely a step. I think it's a smaller step than it seems. I think a few years ago, if you told me you can have 10,000 scans, I would have thought that's amazing. Now, I wouldn't get out of bed for less than 10,000 for the kinds of models we do, so to speak. And we can see that as our modeling architectures get more and more complex, we can make use of more and more data. And the problem is asymptotic. I mean, I think a good analogy is face recognition. If you look at the performance of face recognition models over the years, they were pretty kind of middling for a long time. Then the internet came along and we could generate collections of 1 million images or whatever, and then suddenly they jump up. Now, face is a comparatively far simpler part of the body. And so I think, with brains, I don't know where the ceiling would be, but I think it would be at millions.
So I mentioned that I have one more question. I mean, could you say that the types of resolutions that I'm after in human brain imaging, much higher resolutions, and then taking in your comment before, where you were saying that, if I have resolutions like that, my models get way more complex and way more dimensional, then actually high resolutions such as that are recursive dimensionality for you. They actually exponentially increase the number of scans, the number of subjects, example data points that you would need?
They would, that's right. But they also help recover what the underlying organizational structure is. So when it comes to learning generative models of the fundamental organization of the brain, they might actually help it because they may supply, at the cost of operating at high resolution, signal that reveals the underlying structure of the data. I mean, when we think about how a brain is generated in the first place, how it arises developmentally, it should be compressible. It should be reasonably compressible.
Could I ask a question here? Sorry, Prakash, don't want to interrupt you, but this is a piece of a really interesting discussion, which is actually making links with the first session we had today. And I don't know if Bill is really here or sort of or just here by avatar, but no, he might really hear. So In terms of what would
doesn't
be,
mean I have something intelligent to say, of course.
oh,
But
well, well, I'm hoping he. I'm sure you always have. What would be gorgeous in this discussion, if what were true functionally and medically could be mapped onto what Bill and others this morning were finding computationally. In other words, there's scaling, or some sort of. There's we can dial up your resolution, and that at least the neural net seems to mean something functionally, whatever your coarse graining. Now, there are problems there because, as we remember with Bill's talk this morning, he didn't coarse grain spatially, he coarse grained by correlation connectivity, and that's not quite the same thing. But what we didn't discuss this morning is this chain. So if it is true that there is some spatial correlation to the correlation connection, and if there's some meaning to the structural or informational, the correlative scaling of the brain on any scale, and given that the brains map onto what given that the state of brains maps onto what brains do, then the question is, what is the scaling in terms of brain function? And if you know that, I mean, so here's a beautiful example. If we just take the visual cortex, so Hannah might be able to help us here. It would be gorgeous if a coarse-grained model of the visual cortex gave you a simply coarse-grained, blurry picture. And if you saw all the neurons in tiny detail, you'd understand the sharpness of the full sharpness of the picture all the cones were seeing. And if you only had a micron or multi-micron or a millimeter resolution of NMR, then that would be equivalent to understanding what the patient was seeing in a fuzzy sense. Now, that's just the visual sense, but that gives a handle on what I'm trying to say. What do we know about how the coarse-grained function of the brain, or indeed a neural net, maps onto its scaling structure?
I would say that I think the problem is actually the visual cortex is not a very good archetype of the rest of the brain. I mean, it's obviously very heavily studied because it's so beautiful, it's so wonderfully organized, because you've got this fabulous way of accessing it through the visual, through the ease of parameterizing the visual. But when you look at other parts of the brain, other functions of the human being, for example, behavior, motor behavior, then it's very difficult to uncover that low-level organization. And in particular, what is missing is the mesoscale. We have the macroscale, we have the microscale, but we do not have the mesoscale. We don't have the organization across the full spectrum of scales that one needs. So I would love it if we could build models of the kind we heard about earlier at the whole brain level. But I doubt it will be quite as rewarding as it has been.
Well, it was just the hippocampus, wasn't it? So I might ask Bill, in terms of the function of the hippocampus, what can you say if you've blocked your neurons to 256 neuron blocks? Does that give you a coarse-grained picture of hippocampal function?
So we do know that right, if you're in the hippocampus, you know that the information is, there's supposed to be information about place, about location of the animal, and there is, of course. There's a lot of evidence that there's information about other things too, harder to get at. We do know that in the coarse graining process, the sort of coarse-grained superneurons or coarse-grained neurons that we create continue to carry place information. And in fact, the fraction of the coarse-grained neurons, which are place cells, is larger than that of the cells that we started with. So it's as if somehow in the blocking together, there's some, it's almost like there's some purification of the place information. But we don't know whether that's really important or not. But it's certainly true that information is preserved, right? So for example, if the coarse graining process immediately destroyed all of a piece of information that we know was there and we know was important to the organism, then one would legitimately believe that this coarse graining is a terrible idea, and that doesn't happen.
Right.
So there's a hope that your description coarse grain description of the brain's activity gives you a coarse grain description of what it's doing. That could still be true, but that's a project, right, to show that that's true.
Sure, but I mean, the medical consequences are potentially optimistic, because it means that if that Prashkev doesn't have to wait till MRI can identify every neuron in a patient's brain at that resolution.
One should be a little careful about scale, right? I mean, what if you're recording single neurons? What you call coarse graining might be what somebody who does functional imaging calls the most microscopic, right? So, and I certainly haven't thought that through carefully.
And the other thing that we haven't talked about at all is how it is that you interpret what these images are saying. And you can only do that in what it is that you can see in the individual or the patient, as it may be. So, and what it is that you can see is something that is resultant of a past history. So you've got to understand that too. And you've got to understand the behaviors that are changing that brain-body behavior axis. So it's a snapshot in time that you're trying to interpret in this space of knowing what's going to happen in their future. So the kinds of predictive models that you're thinking of aren't just anatomical, but they're chemical as well. And they're also behavioral too. So it's trying to put all of that together into a predictive model. This has huge ramifications for pain, for example, and knowing what to do in terms of not just predicting who it is will react well to one treatment over another treatment, which is that prospective modeling that you might do, but also in predicting who's going to age healthily, for example, or not, as the case may be. And it changes at a rate of possibly minutes, but some people might want to challenge me that it's quicker than that.
I was going to switch gears to try and get Hannah to talk to us a bit. And think of, and David also, because they're actually looking at single neuron, really, and their interactions. But the thought I had with Hannah, and I mean, it's well known, but the optical stimuli that you're providing to individuals are things that they've never experienced before, because the environmental optical image isn't it's aberration limited, not really diffraction limited. So stimulating individual cones and perhaps even neighboring individual cones, you're giving them access to things, a super resolution, if you like. So you've got a series of experiments that you can do that might indicate whether super resolution is accessible, given that it's not being seen by the participant ever before, both spatiotemporally. So is a Reichardt detector, which is a motion detector, possible with two neighboring cones, for example, and things like that?
Yeah, I mean, those experiments are beginning to be done. So obviously the first experiments with sort of cone stimulation were just stimulating a single cone, but now collectively people are beginning to look at pairs of cones and temporal delays between them, etc. And you get perfectly good visual percepts from these things. So I think that's one thing that, I'm biased, but it's one reason why I like working in the retina. So one of the ways that I make the problem tractable is to say, okay, I'm going to limit the stimulation. So you stimulate a cone that activates several that'll drive more than one neural circuit. But it also goes all the way to driving behavior. And so we've got a whole system level response to that stimulus as well. And we've got eye movement level response to that stimulus. So it's a different way of thinking about scale, I think. It's how that very small scale input then propagates through a whole system that you haven't messed around with. It's a full intact system. I think one of the things that's clear in vision is that we know a lot about the underlying circuitry and subcircuits. And one of the reasons why we can dissect those subcircuits is because we seem to understand something about the primitives of vision, as it were. So we seem to know, we've worked out what particular classes of ganglion cells seem to extract from the visual image. So I think that makes it more tractable. But I'm not sure I completely agree with the idea that that's not scalable to other brain areas. I think we just don't know the primitives for other brain areas. But maybe that's a hard task to search for those primitives.
But what is the primitive for social interaction?
Yeah.
I mean, this is the problem. If you take, I don't know, something like frontal pole, how do you begin to think about how it might be organized?
Yeah.
When the deficit that you get following damage is actually so hard to pin down. It's catastrophic, and yet it's so heterogeneous that you can't even model it behaviorally, let alone neurally.
Yeah.
But we've got to try, of course.
Yeah. I mean, I haven't got a good response to that, except that presumably that's at one point how people thought about vision as well. We haven't always known about centers around receptive fields or whatever. So
Sure. But it seems to me that there is a reason visual neuroscience has been so much more successful than other domains.
Yeah.
It's because it is more tractable to the kinds of study and the kind of modeling that we can do. I don't want to say it's simpler. It's not simple. It's incredibly complex, but it is, it is tractable. I think.
Yeah, we perhaps have a better language for describing it as well. I mean, the mathematical description of visual stimuli has served us quite well.
Yes. Yeah.
Can I just ask a question? Just coming back to Tom's question, something Peshka was mentioning hierarchies. I was wondering if there's any relationship in your appreciation of the functional architectures between the notion of a hierarchy, as we've seen in terms of visual cortical hierarchies, starting with the retina and the apparatus of the renormalization group. Is there some way of using what is, to my mind, a cardinal architectural feature that transcends many scales, which is a hierarchical organization of connectivity and message passing in the brain that could actually be taken right up to understanding social interactions from the level of interpreting visual sensations at the retina. Is there any way that you could take hierarchy as a frame to talk about different scales using things like the renormalization group? Because that strikes me as a useful way of bringing a physics of structure and scale invariance to the empiricism that we're all contending with at different scales.
Well, I mean, I would say, again, I think it depends on the specific task. If our objective is to come up with the most faithful description of the operation of the brain, as far as it's possible to present it in the general, then the most graceful way to model it would be hierarchically, so that you can then maintain some kind of intelligibility of your description, while being excused for not being able to predict what happens in the specific individual in front of you. By contrast, when it comes to something such as the development of a machine that seeks to come up with the best answer about who needs what treatment or what will happen to a particular patient, then it may be necessary to come up with a different formulation. It would be, of course, very attractive if it turns out that the hierarchies that we've been able to observe within macroscopic, relatively simple models do indeed check out when one descends to a final level of description. That would be wonderful, but it might not be so. And perhaps we need to think about how we can take these two different tasks and find some kind of happy medium between them. I mean, ultimately, a perfect inferential model is going to be perfectly predictive. But at the moment, we have a situation where highly predictive models are not necessarily that illuminating inferentially, and vice versa. At least in the domain of focal brain injury.
And I think Carl mentioned the retina specifically. I think so. I mean, Hannah can put us right on this one, but I think there's a sense in which the retina is an example of tissue that sort of renormalizes itself, or at least in some animals, because signals from a primary layer are grouped together in an X layer, and sometimes that grouping is itself hierarchical. So, is that, Carl? Is that what you had in mind? Is Hannah? I got that idea that there is a.
Yes, and things like the canonical microcircuit as being a motif that is actually replicated at many, many different scales. So yeah, that's exactly what I had in mind.
Tom, I'm conscious of time. And
me too.
I'm assuming I'm handing over to you to close the meeting.
You can do.
Is that? Oh well, I mean,
that's really it. Other than from Tony and myself and all the other chairs, an enormous thank you to all today's and particularly this afternoon's second session speakers. Thank you so much.
Absolutely, thanks very much.
It's been fantastic.
Really great.
Lots of ideas, and we can practice using the Wonder this evening. It's an informal session, as I mentioned earlier. Tomorrow we'll have a little bit of work as well as play. Thank you very much. Do join us tomorrow, and I'll see you in the Wonder Bar in 3 minutes' time with a beer. Okay. Bye.